Symposium is on evolutionary genomics in agriculture, environment, biodiversity conservation, and health for Africa 2026. So welcome all. We'll start with our first presentation, and I'm so honored to meet Professor Grobler again after a long time when I was a PhD student in South Africa, and he took us around in Kruger National Park. He inspired us to come back and start the lab. And I'm so happy James came up with has progressed, and in the University of Eldoret, we are running a lab that we are going to discuss at the end of the 3 days. So Professor Grobler is a head of Department of Genetics at the University of Free State, South Africa, where he also leads research on population and conservation genetics research group. And previously, he has worked at the University of Limpopo, and he was adjunct professor at Department of Fish and Wildlife Conservation at Virginia Tech University in US. He has supervised many students, including James and many others, has many publications in accredited scientific journals. There is so much to talk about Professor Grobler, but in this symposium, Professor Grobler will provide an overview of current conservation issues, explore how genomic approaches can advance conservation, and by deepening our understanding of population genetics processes and investigate pathways for their practical application in the field. We want to welcome Professor Grobler with a clap as he comes to do his presentation. Welcome so much, Prof. We are so happy to meet you and to have you around at the university and in Kenya, generally in Eldoret.
Right, good morning everybody. Should I hold the mic or is it fine like this? Okay, right, so let me do this. I'm going to need many hands now between everything I'm doing here, but we're going to manage. Right, so yes, good morning, and very, very special for me to be here with you in Eldoret, my first visit to Eldoret. Thank you also to the organizers for inviting me. So yes, my presentation today will focus on conservation issues in Africa, and especially my part of Africa. And I will look at the various research questions and what we learned using classical approaches before the onset of genomics, and then look at how the increased amount of data that we get from genomics can add to that. I said that change the answer. It's not always about changing the answer, but about getting more comprehensive answers. Right, so I thought I would start with 1 or 2 minutes just telling you exactly where I come from. So I'm attached to the University of the Free State, as Emily said, which is right there in the middle of the country where the yellow dot is there. There's some views there of the city of Bloemfontein where I live. And much more interesting, if you look at the two at the bottom, that's typical landscape where I live. The Free State is very well known for its open plains and grasslands, but actually in the eastern side, we also have some very, very nice mountains. That's the University of the Free State, the main building on campus. So yes, we're moving towards our 130 years anniversary now, and we have just over 40,000 students. That's where I live. That is the Department of Genetics. Not so nice as the main building. but a newer building on campus. And in genetics, we have various areas of specialization: human genetics, behavioral genetics, plant genetics, fungal genetics, lots of work on forensics, and then our group on conservation genetics. And that is, of course, what I will talk about. So my field is conservation genetics, with a special interest in the application from a management perspective. Right, so what I will do today is to talk about some of the current issues that are relevant in conservation in Africa. And as I've said, we will explore what classical approaches got right, and also where you hit limits when you are using such classical approaches. And then, yes, return or move on to communication with conservation practitioners and how we turn genetic and genomic data into conservation decisions. Right, so what are the kinds of conservation issues that we look at? And if I look at it from a South African and Namibian perspective, there's a couple that always come up. So it's obviously the management of the many small and fragmented populations. We work on hybridization between distinct species, genetic structure within species. We're looking at non-invasive sampling and monitoring of animals, parentage and pedigrees, wildlife forensics, genomic databases for evolutionary and systematic discovery. I don't do too much of that, but of course there are many other researchers in the country that specialize in that from a conservation perspective. And then again, yes, trying to translate all of that to management information. So not all of that is applicable outside Southern Africa, but I think the broad questions are often the same. So the kind of things that comes up in other parts of Africa is understanding population structure across large landscapes and the effect of various developments on connectivity. Yes, transboundary conservation and how political boundaries is a mismatch often for genetic population boundaries. Very important these days, the preservation or conservation of adaptive variation. When you talk about conserving genetic diversity, again, evolutionary and systematic discovery, and also wildlife forensics, of course, in other parts of Africa. Right, so I want to start with some basic background information, because it's very different from what a lot of you will be used to place it into perspective. So those two pictures, to me, symbolize a lot of what we do with wild animals in South Africa and Namibia. And it boils down to really intensive management, artificial management of wild animal populations outside of the formal conservation sector. So totally outside of national parks and reserves, there's a very large private game industry where a lot of this happens. So if we look at the conservation landscape in South Africa, from a public perspective, we have about 6 million hectares under national parks. And the private amount is actually bigger than that. So there's around 16 million hectares that consists of private properties that intensively keep and manage wild animals. And there's a big reason for that, and that is because of the somewhat unique situation where we have private ownership and monetary value of game. So kudus and eland and many other animals are bought and sold like sheep and cattle, and it's a huge industry. There's probably 9,000 or so farms in the country that operate this way. So the importance or the importance to us as conservation genetic scientists is twofold. First of all, many of these reserves and game farms are, of course, small and isolated, whether they are private or not. And then translocations, and there's a bit of irony in that one. South Africa is undoubtedly a world leader in translocating animals. We move anything up to and including rhinos and elephants and put them on Boeings and fly them across Africa, which is great from a management perspective, but of course it does mean that all kinds of things are mixed and we should look at the implications. Right, so I'm going to talk then through a couple of the aspects that I've mentioned, and I will see how many of them I can cover. So we start with the basic one, which is the management of small and fragmented populations. And there we've done many studies on a variety of antelope species, as well as lots of work on rhinos. And I will talk about some of that. Yeah, this one I hardly have to show, but just a reminder, why are we concerned about small population fragments? It's all about the classical processes of bottlenecks and founder events that happen on these small localities, especially founder events, starting populations with small numbers of animals. So let's look at one case study there, and I've for each one of the projects that I present, I've given the lead researcher in my unit or collaborations that's doing it. So this is a project by one of my PhD students, and he's specifically looking at its animals from an already bottleneck population, and that has the lowest level of diversity. And then we look at Doornkloof, started with only 14 individuals, currently at 109. And if you look at diversity, it is right up there with the control population, which is very, very confusing. And you get the same thing if you look from at heterozygosity. So largest or highest value in the control population goes down as the founding or the number of founders becomes smaller and smaller until we get to Doornkloof, and it's back at the control levels despite starting with 14. So you can see that cannot really be. So there's a very interesting or simple explanation from it. If you look at Doornkloof, the reserve there in green, it is surrounded by a very, very large privately owned piece of property. And that the border is somewhat porous with lots of rivers and so on. So there's obviously animals moving from Doornkloof into, or from the surrounding property into Doornkloof, which is giving it that unintentional gene flow and much better levels of diversity. And interestingly, that large private population got its hemsbok from the same place that we used as our control originally. So that gives Doornkloof a nice level of genetic diversity, a little bit by accident. So we're doing very, very similar projects on a range of other species, notably Hartebeest and eland. And we're sort of at the point where we're trying to do the same kind of study, but moving to RAD sequencing so that we can add runs of homozygosity as a measure for our diversities. Yeah, we're also doing lots of similar work on rhino populations. Rhinos in South Africa are very interesting. There's around 14,000 of them at this point in the country, but depending on which version of the history you look at, they went down to somewhere between 20 and 200 individuals in the 19 or the 1800s. Any one of the two figures is not a good thing. And there's been, well, they recovered very nicely to the current 14,000, maybe more. Could have been a lot more if it wasn't for poaching. And there's many translocations around populations, but you have to forget that ultimately they all come from the same place. They're all from the survivors in KwaZulu-Natal. So yes, we're working on them quite extensively. There's just a field work shot of rhino in the process of being darted. Yeah, and if we look at the results, so we just a little bit of that. We use as a yardstick, we use some work done by Joshan Moodley, and he established levels of diversity in some fairly representative populations. And if we just look at let's just look at the expected heterozygosity. So he set a range of about 0.44 to 0.45. And if you look at the privately owned populations that we've been working with, they're in the same league and sometimes even a little bit higher. And it goes back to founder history. People that do reasonably good genetic management do not stick to a single founder event, but substitute numbers over time, and you can see the positive effect of that. I want to say something about why we should be interested in studies of diversity, because people tend to be a little bit fixated on visible defects, and you actually see very few visible effects of losing genetic diversity. That's horn deformities is one that you do see from time to time, but that gives a false sense of security, because a lot of what you think is populations that are genetically doing well is really just a lack of records and a lack of careful observation, and you do not really know what is happening in terms of fitness in those populations. And there's a very nice example in South Africa that can really give you a totally wrong impression. So that's a Himalayan tahr up there, and they used to keep a couple of them in Cape Town, just below Table Mountain, which I think all of you know, in the Grooteskier Zoo there. And then at some point, two of them escaped, a male and a female, and they ran up Table Mountain, and they did extremely well. And by the time conservation authorities started doing something about them, there were at least 550 of them. So that sort of situation is often used as an argument for why should we not really worry about inbreeding. That's the worst possible scenario, one male and one female, and they went up to over 500, and they're doing well. So they've actually removed hundreds of them, and they're still not all gone. They're still, but of course we should not be deceived by the fact that they are doing well, because they're only probably doing well because they're living in a very stable environment. There's very little to stress them. There's certainly no large predators up there. So even though the numbers look good, they may not be as successful as we may think. Right. So to wrap up the diversity work. So what classical conservation genetics I think got right is well-established tools for quickly getting a level or an indication of the level of diversity in a range of species, and using that to determine various things like bottlenecks and effective population size. But of course, we can do a lot better of that, or than that. So the way forward in terms of diversity studies is certainly more genome-wide work. And as I've already mentioned, doing, for example, runs of homozygosity from SNP data. Of course, using more markers gives us simply more data points in species with historically low diversity. And again, the adaptive significance, genomics allows us to move towards the so what factor. We've lost diversity, but what now? I just want to quickly talk about these guys. So there was a phenomenon in the South African game industry that was extremely popular for a while, and then it died out, and I see it's starting again. And that is extremely intensive breeding and selection for different colored phenotypes. So that, if you do not know, is a springbok, and that is a very widely distributed animal in the arid parts of the country. There's some other springboks. So if you look at the top there's a white springbok. If you look at the right-hand side to the also to the top, there's a black springbok and a copper springbok. Here at the bottom is also some white springbok. So a very large industry actually started as a result of this with very intensive breeding. And we sounded lots of warnings about this, because you're now creating founder events at a very serious level, because you have to think where these animals come from. There's no genetic manipulation involved here. This is just low frequency genes occurring in populations. And now you take the these ones that is 1 in 100,000 and breed with them intensively, and there's quite a bit of risk, I would say. But we also did get a bit of a surprise. So one of my former postdocs, Riel Kutzer, he took about 60 or so of the different colored Springbok varieties, regular, black, white, and copper. And at that point, we just ran them on a bovine 50K chip, which works sometimes. So we got about 3,700 snips that worked from that chip. And if you look at the comparison of levels of diversity, what the level was in green in common Springbok versus what you get in the intensely managed ones, there wasn't really a difference but at the same time, it's a bit of a false image there because he also sampled from various localities. So the numbers there for, let's say, black Springbok represents more than one place. That is not to say that on any given farm, it's going to be the same. But yes, ultimately a bit more diversity than we thought. So yeah, we're lucky we used heterozygosity here rather than runs of homozygosity because it's a cross-species application. I think conceptually, runs of homozygosity is not entirely appropriate because you don't know what happens between these data points if it's a cross-species application. Right, then next one, interspecific hybridization. So that, as you can see, is a cross between a zebra and a horse that I photographed in the Karoo. And that's fine. There's not a lot of that around. It's a novelty, but there's lots of hybridization going on that's not novelties. So yes, Prof. Tiedemann, you will remember some of this. So there's a couple of pairs of species that are very prone to hybridization, and the ones I've spent a lot of time working on are black wildebeest or gnu and blue wildebeest. And you can see if you look at the distribution, they're opposites. The black wildebeest endemic to the grasslands of South Africa, the blue wildebeest everywhere else, right up to this part of the world. Move them, bring them together, and you get hybrids, and they are fertile. There's another example with zebra. We have plain zebra, and we have Cape Mountain zebra. You can see the phenotypic differences between them, totally separate distribution areas, but mix them, and you start seeing hybrids. There's a very similar situation in Kenya that was a natural hybrid zone, not translocations, between plain zebra and Grevy's zebra, also involving hybrids. And then the tough one, Blesbok and Bontebok, they are very close together. I can't even always tell the pure ones apart, so what about hybrids? But again, Bontebok and Blesbok are kept together when they should not be kept together, and you have hybrids. So maybe I should first just say why do they hybridize? And there's a number of reasons, but the big one is moving things around beyond where they used to occur, and they start mixing, and there's a reason why people move them around. If you have a game farm and you want tourists to come and view your animals and pay for it, you want to have species diversity on the reserve, and you start bringing in things. Size and habitat diversity has a big effect. Just because you have black wildebeest and blue wildebeest together does not mean that they will hybridize. If it's a big place, you could get away with it. If it's smaller, then maybe not. And then very important, sex ratio and the availability of species-specific mates. If that is wrong, hybridization is of course going to occur. And like I said, this also happens here and there in natural hybrid zones, including zebras here in East Africa. Why are we concerned about this? There's a number of reasons. Ethically and legally, of course, we have an obligation to keep things in their pure state. There's possibilities that hybrids can be more successful than pure animals and outcompete them. And the one that I think for us is the biggest motivation, in all of these cases you have a rare one and a common one. Black wildebeest, there's maybe 17,000 on the planet. Blue wildebeest, there's 1.5 million. So you don't want the black wildebeest to be swamped by the others. Same with plain zebra, same with Bontebok Blesbok. There are hundreds of thousands of Blesbok and much, much fewer Bontebok. You don't want to lose that purity. Right, so there's just a nice picture of two first generation F1 hybrids, and you can see it in the horns. If you look at the pure ones at the top, blue wildebeest, nice wide horns, black wildebeest goes forward, hybrids somewhere in between. So we did this, we've been doing this for a long time, and we did some smaller studies before, and then in more recent times we actually did this at a very large scale where we sampled almost 500 animals, so large numbers of pure black as well as pure blue, as well as hybrids and putative hybrids to see what we could get. We had 24 loci that we used there. That's the overall outcome of what we did. So that's, for those of you who know structure software, that's just structure turned on its side with 500 data points. At the top you have pure blue wildebeest, at the bottom black wildebeest, and a zone between them. So if we zoom in a little bit on that zone, you can see that's where it gets interesting, where you have either 50-50 hybrids, or if you start getting back crosses, because that is what happens, you have first generation of hybrids that you don't pick up, or you do pick them up and you take out one of the species, and they start back crossing with the pure animals, and that's where it gets interesting. So you can see some animals there that shows lots of admixture in their genetic makeup. You can also see it from levels of diversity. So if you look there for black wildebeest, if we just let's just look at the number of alleles. For black wildebeest, we got on average 1.7 alleles per locus. For blue wildebeest, we got 2.6. And if you look at the hybrids, it's somewhere in between. A good indication that there's hybridization happening there. Right, so the interesting part with all of this is being able to detect it long after the fact. So seeing a first generation wildebeest hybrid is not so difficult. But as I've said, if it start back crossing to the one side with each generation, you can have a 50% dilution of the effect. So this is based on a very simple diagram based on some very long simulations that we did to show what happens on average with hybrids with each generation of back crossing. And you can see that if I put in that little line there, you very quickly reach a point where it's extremely difficult to identify something as a hybrid. You could, of course, ask the question, does it then still matter? If you struggle to detect it, does it matter so much? But from a conservation perspective, the feeling is that it does. Pure is pure. Somebody did some calculations on what it will take, the number of markers that you need to identify hybrids as backcrossing continues. And you can see it goes up very quickly with each generation of backcrossing. And you can see where this is going. You run out of options with classical markers and you need something new. So for that reason, my former student, Andri van Wyk, who's now a postdoc, she's taking this further, and she's now done some genome sequencing for 15 individuals representing pure black and blue, and some hybrids in between. And we're going to use the results in two ways. First of all, she's identified a panel of 240 SNPs that we will now use. It's actually not so many, but it's still a lot better compared to what we had before, so that should be very valuable. And of course, once you have genomes, you can go a lot further and look in terms of ancestry informative markers, gene variants at various places that clearly separate the two species from each other. Right, so yes, again, to summarize, what classic conservation genetics got right was to give us practical models to detect early generation hybrids, but genomic approaches will give us lots more loci to detect advanced back crosses, and it will hopefully give us some data on ancestry informative markers. Okay, we move on to something completely different, vervet monkeys. So we've done really a lot of work on monkeys. I think around here you also call them savanna monkeys or African green monkeys. So we've gone through a whole stage or process with monkeys from simple mitochondrial DNA work to single genes with adaptive significance to genomic work. And we've probably sampled more than 1,000 monkeys across Africa in the years that we did this project. And this actually started in an interesting way. It started with primate sanctuaries, that because people like to keep monkeys as pets, baby monkeys, but it's only a nice pet for a while, and then it becomes a highly destructive animal, and it's also illegal. So they end up in sanctuaries, and it started when conservation authorities was getting worried about all the monkeys that's being mixed in sanctuaries, and you don't know whether you're actually mixing genetic variants from different parts of the country. So we did this initially using mitochondrial DNA, and the results were quite nice. If you look at that map of South Africa from a biomes point of view, you can see some of the main features of the country, the savanna areas all around the sides, the grassland in the middle, and the coastal forests around the sides. There's many others, but we'll just talk about that. And we got the idea that monkeys over a very wide area in the savanna areas that I'm highlighting in blue there now are actually genetic very, very similar, which makes sense. The ones in my part of the world, in the Free State, are quite a bit different because they're isolated from the rest. And then the ones around the coastal area, the wet areas, is also a bit different. So that was a good start to the project. And then this project really took off. So we first started looking at genes with adaptive significance, and we looked at toll-like receptors. And there we looked at diversity in the toll-like receptors in populations living in arid versus much wetter conditions, on the assumption that in wetter conditions there will be a bit more stress that the monkeys have to respond to, and that you could see more diversity there, and that is actually what we saw. So more diversity in the toll-like receptors in the wetter parts of the country. And then this project really took off because then we started collaborating with UCLA and what ultimately became known as the International Vervet Consortium. There was a reason why the project took this direction, and that is because of something called SIV. SIV is very closely related to HIV, and monkeys have it. In the ones we've tested, more than 50% of the population will carry the SIV virus, but of course they don't get it. They've lived with it forever, and they have co-evolved and they have resistance, and of course there's good reasons to look at why that is so. So we sampled monkeys, and ultimately 163 of them full genomes was generated for. At that point, it was the biggest genomic data set for a non-model primate organism that was developed. So 163 full genomes. And then we did various things for it, and at that point, Prof. Hannes Vardal also became involved in it, and he did the analysis of all of those genomes, and ultimately we got a very nice paper out of it. So yes, we looked at the taxonomy of vervets in Africa and the recognized subspecies, and of course went and confirmed all of that, but now using not a little bit of mitochondrial DNA, but full genomes, and got a very nice indication of the classification and relationships of vervet monkeys. And what was interesting there is that there was a very strong signature of selection involving resistance, exactly as one would expect. So of course there's many other reasons also for the patterns that you see, but certainly adaptation in terms of resistance against viral infections played a significant role in their final taxonomy. Right. And then something very interesting happened. Then COVID came along, And it, as you can see here, it had some effects on our fieldwork, but we persevered. And then Christopher Schmidt at Boston University, who was part of this project for a long time, started asking the question, what about monkeys in captivity? I mentioned a while ago all of the vervet monkeys in captivity in sanctuaries. So if you have COVID in humans and there's contact between humans and monkeys in confined spaces, what's the possibility for zoonotic transmission here? And this is the nice part about genomic work, that the whole genomes were already there. It did not take any new sampling or even sequencing. It could just be another remining of the data, and he looked at ACE2 primarily. And without now going into too much detail there, what he basically found is that he looked at binding sites on the cell surface where the SARS virus can potentially attach to, and he looked at variation there, and he compared it to a variation in humans, and of all the sites that he looked at, more than 20, there was only one where basically the variant existing in the monkeys would not allow the SARS virus to attach. And for all the others, even though there were small mutations, it was missense mutations. So it appeared from a analysis point of view that vervet monkeys should be almost as susceptible to COVID as humans are. Yes, by then the epidemic started going down, so we did not pursue this further. Right, something about genetic structure still in other species. So we're doing a lot of work on kudus from various perspectives. So our current results indicate that in kudu there's definitely two clades, the eastern and the western clade, established once again using traditional markers, and to take it forward from here, it is about, and we've just started some Oxford nanopore work on them. It is now on what is the reason for the divergence, and do we pick anything up in terms of adaptively significant genes. We did lots of work on giraffe. Giraffe are very interesting taxonomically. People are forever debating the taxonomy of giraffes. And it gets very complicated, but in Southern Africa it's not so complicated because there's only two subspecies. There's the South African giraffe and the Angolan giraffe. Of course, it's not really so nicely limited to countries, but we call them the South African giraffe and the Angolan giraffe. And you can see there where they occur. So very, very interesting, again, in my part of the world, because it's a grassland, there's not really supposed to be giraffe, but there are hundreds of giraffes that were introduced. So we looked at their origin, and it's interesting. If you look at that haplotype network, what you see there on the right-hand side are the pure South African giraffe populations, more here in the middle. The dark green is Angolan giraffe, and the light blue haplotypes are ones that were identified from private properties, and you'll see that it spans everything. There's Angolan giraffe and there's South African giraffe in private South African populations, and that makes sense because over the years, once again, many translocations from elsewhere, adding giraffes that were not here from the start. Right, so we're continuing this project, looking at diversity in small giraffe populations. That is very, very relevant because giraffe populations are very often very small. People keep all kinds of animals on game farms, and then giraffe, as an afterthought, there's 5 or 10 giraffes, so lots of small populations. And again, we should look at the adaptive significance of the taxonomic patterns as well. Right. So yes, for long, classical approaches were the standard for phylogenetic analysis, but of course now it's different, and we can look at very small scale differences. Especially if you look back at those monkey genomes, there's a gold mine of information there. And as I showed with the COVID result, describing patterns and generating the genomes, the first time is just the beginning, and it's really actually the start of lots of new things that you could do. Right, I'm going to go through the rest, I think, a bit quickly. Time is probably slowly running out. So we're doing quite a bit of work at this point on non-invasive DNA-based census techniques, and that is again in Doornkloof Nature Reserve that I like very much to work on. So what is that all about? If you want to count black wildebeest, it's easy. They're out there. You do an aerial count, or you use a drone, or you drive around and you count them. If you want to count kudu, it's a little bit different. If the habitat looks like this, and that's this way kudu like to occur, then it's not so easy to count them, same as that one. And kudu are actually still on the easy side of the spectrum. You get many other animals like Inyala and your small antelopes that are extremely difficult to count in terms of a census. So I have a student working there on the reserve, and he's using spatial mark recapture to estimate population size. And I think many of you know the principle, especially in aquatic systems. Some of you may have done this as an undergraduate student, where you mark Daphnias and release them and get the idea of the population size. So it's all about sampling a population once, marking them, releasing them, sampling it a second and a third time. The only advantage here is that you don't actually have to mark them, because they've marked themselves. You use their DNA profiles. So yes, what my student does is he walks transects like that through typical kudu habitat, and you have to repeat it a number of times to get statistical strength, picking up fecal samples. There's just a view of some picking up of samples. Yes, and in his initial pilot study, he collected 137 samples, of which, unfortunately, quite a few were not kudu. That just happens in the early stages of a project, but many, 114, were kudu, and we could differentiate 91 unique multilocus genotypes, and then we applied mark software, and it gives you, based on how you got to this, it gives you an estimate of population size, which we determined to be 161, which was actually quite close to what is known from counting. So that's interesting work, and of course you can take it further, because you can also get basic demographic informations. It's easy enough to do sexing of animals with the right marker. I have to say the one that I would really like to be able to do from such work is age determination, which is not impossible, but it's not easy. I actually have a PhD student, Louis Leclerc, who finished 2 years ago, and he did age estimation on cheetah, DNA-based age estimation, and he used methylation of DNA of various regions, and he was ultimately able to age a cheetah within 18 months based on a DNA sample, which is nice, but it's lots of work, because you have to calibrate your model with lots of animals of known age before you can do it. But still, I think adding aging or age determination to non-invasive sampling will be a very valuable addition. Right, we're doing some non-invasive work on porcupines. Porcupines are very nice because they leave the samples for you, and you get high quality DNA out of quills. So I have a student that's working on that, sampling them in various parts of South Africa. I would actually be quite interested to talk to somebody in this part of the world who also want to sample porcupines around here, so that we can compare. So talk to me if you are interested. And yeah, we also do fecal samples, but the quills are a lot better. We've dabbled a little bit with aardvark. Aardvark, as you can see from the quality of that picture of mine, is not something that you see very often or very long. I've seen very few in nature over all the many years that I've spent on field trips. So yeah, we're playing with some ideas on and now this is moving closer to environmental DNA. To sample them in the places where they live and where they feed. And of course, the next step to that is true environmental DNA. We don't have any active projects on that, but we will hear about this on Wednesday. Right, so yes, classic approaches gave us data on species presence and sensor size and some demographic data. Genomics takes it all forward, and we go from what species is here to how many species coexist in this ecosystem. Very briefly, something about parentage and pedigrees. So just going back to the white rhinos, we're doing a lot of work on that to confirm pedigrees. And there's an interesting thing when it comes to rhinos and pedigrees, because you have this dichotomy. You have rhino bulls that are incredibly successful, and you end up with a situation where half of the calves on a property are brother and sister. And it looks very good from an ecological or demographic perspective to have such successful animals, but of course genetically it's the opposite, and you're reducing your effective population size if you have so many related animals. So there's actually one property that we're working with, a very large rhino owner, that actively try to manage this and at the right time remove 2 successful animals from the equation. I'm not going to say much about the rest here, except to say that we also using some genomic approaches at this point to identify additional markers to specifically track male-mediated gene flow. Still in terms of parentage, another student of ours, Michelle, she worked on cheetah, and she compared microsatellites and SNPs and how effective they are to reconstruct pedigrees, and she had a nice data set with known things. So she had 76 cheetah available, of which 56 were of known families and another 20 were unrelated animals. And she used both microsatellite markers and SNPs that she developed through rat sequencing to see how effective from genetic information you can reconstruct the pedigrees. And yeah, obviously there was a difference. From microsatellites, quite a big 24 loci panel. If both parents were included in the data set, she could get it right 18 out of 22 times. That's not too bad. And if only the father was present in the data set, she could still get it 17 out of 22 times. And with her SNP panel with 218 loci, she could get 22 out of 22, 100% success if all the animals are indeed in the data set, and very close to a perfect score when only the male information was available. Yeah, briefly something about wildlife forensics, which is very prominent here. When you talk about wildlife forensics, there's always 3 questions. What species is this? Where did this animal live? And what specific animal did it come from? And the first one is comparatively easy, species identification. You can do with barcoding, so it's not difficult to do. So a couple of years ago, we had a project where James was also involved. So there were some illegally kept Asian walking catfish in South Africa. It's a highly invasive species, very similar to African catfish. And again, I think if they hybridize, it becomes a lot worse. So there was some suspicion of introduced walking catfish in South Africa, and we quickly sorted that out, and we could show that the suspect fish were indeed Asian catfish and not African catfish. The other 2 questions are a lot more difficult, and it's more difficult for various reasons, but the big one is databases. If you ask the question, where did this animal live? You can only answer that question if you have databases of all the places where that kind of animal lives. And that exists for very few species, and very often you have to almost create a database from scratch for a new species. What specific animal did it come from? There you can use a lot of the principles of human individual identification and also apply that, but again you need good databases. Right, so in terms of classic approaches, species ID and individual ID worked very well. Genomic approaches will certainly help us to get much more precise geographic assignment if you have the necessary databases. It's definitely going to be a huge advantage in species with low diversity, where you don't get enough variation with other markers. And there's also some benefit in terms of using mixed samples. I want to quickly tell you about this project, and then I'm almost through. It's a bit off topic, but I think you will enjoy it. So I have another student, Nte Mankwana, who just finished his MSc 2 years ago, and we published his work in Forensic Science International. So we moved from tracking animals to tracking people through touch DNA. Now, those of you who are not familiar with touch DNA, it's very big in forensic science, and the idea is that everything you touch will leave a trace, and you can get touch DNA from it. And then we asked the question, but why not also a rhino that's being touched? So we actually did some nice trials on tranquilized rhinos. So the idea was to recreate the scene that would typically occur where a poacher is involved. If they're going to cut off the horn, obviously they're going to hold the head, hold a few other places. So we swapped we first made people touch the horns, and then we swapped them, and we also did something that's known as tape lifting, which is exactly what the name says. You put a tape on the touched area and get your DNA from that. And yes, ultimately it's definitely not foolproof, but we actually got lots of usable human DNA profiles that way that can certainly help to identify poachers. Right. If we move a little bit to the pure evolutionary work, maybe closer to the central theme of this symposium. So my postdoc there, Zhao, has been quite active in that. He's a reptile person, and there's one project there especially that's I think is going to give us very good results or interesting results. So he's working on the genus Scylotus, or the burrowing skinks. And the interesting thing about this taxon, of course, is the loss, or the apparent loss of their limbs. So you get the whole spectrum if you go through the taxonomy of Scylotus in South Africa, from having very small limbs to basically a limbless state. So we now have 11 different species of Scylotus that is being sequenced with long-read technology, and then we will start looking at it. I should perhaps also just mention, in this case, we send it to, there's an initiative in South Africa called 1KSA, which is trying to sequence 1,000 genomes, so you can actually nominate species to do it. So we made use of that opportunity and got them to do 11 genomes for us. I think there's also a similar initiative in East Africa to create many genomes. Right, we're doing the same with tortoises. I'm not going to say much about that because I'm out of time, but I do just want to answer or ask a question at the end and make a recommendation. So I do want to ask the question, does more data equal better conservation? And it's a silly question, of course it does, but it's not so silly because more data only equals better conservation if the data gets to the right places. And especially for the students amongst the audience, I think that's something very important to think of. If you look at the big difference between us as conservation scientists sitting here and what happens at ground level, and there's different levels to all of this. I mean, we have us academics, professional scientists sitting here. You have regional managers for conservation and parks authorities, and then you have the reserve managers on ground level. And reserve managers are much more interested, because they have to be, in fighting fires and stopping poaching and rebuilding roads and mending fences than genetic management, and yet we have to get information to the right people. So I've just wrote down a few things that I think are important with that. If you evaluate, and I work a lot with conservation professionals, if you look at it from their perspective, it's very interesting. How many of them read scientific papers? There's certainly a hardcore of park managers who read conservation papers, but many also do not. So what we publish in higher impact journals is not always going to reach who it should. They do extensively read synthesized papers, distributed as reports. They rely very much on workshops and conference presentations, but especially in-house workshops. Very interesting. Every reserve manager I've ever spoken to has at some level an appreciation for genetic diversity, and that's just because of what we know from agriculture. So there's an appreciation for the concept, and there's also a growing awareness of the whole concept of geographical differences and not mixing things when we should not. And there's a very strong culture of following both national and international conservation guidelines in terms of conservation. So what I think we should keep in mind is really to report back to people on ground level. And I'm just as guilty as that. It is very often easy to forget where your samples came from once the nice paper has appeared, but I think we should make special efforts to go back to the reserves where we work and give in-person report backs and organize workshops and so on. What is very important is to reduce techniques and all of our nice statistics to values and comparisons that make sense at the relevant level, to integrate genomic and genetic data into ecological contexts, emphasize definitely what can go wrong in terms of if you get it wrong with both diversity and differences. And then, of course, take it further and formulate management strategies. And I have to say there's a lot of that going on in South Africa with management strategies on forums combining all the necessary stakeholders. And yeah, this one, it's interesting that after all of these years, it is still a challenge. Every reserve manager, when you talk to them and present results and tell them about the dangers, will ask you for a specific answer. How many of what should I introduce, how often, to not lose genetic diversity? So, yes, I think there's certainly advanced software that can be used for that, but I also think there's a bit of a gap here for something very user-friendly. We did something in that vein for the white rhinos. So for a large rhino owner in KwaZulu-Natal, we developed well, it's actually a simple spreadsheet, but they can put in variables like how many of each sex they put in at what intervals and what the level or the change on level of diversity is going to be. And it is surprisingly easy in a way, in the sense of how little is needed to make a big difference in terms of bringing in new individuals. Right, so that brings me to the end. So all that remains is to say thank you to everybody, to you for listening, to the society for sponsoring us, definitely to James for all of his hard work, but also to my students and postdocs and collaborators whose work I've included in the presentation. So thank you very much. I'm okay standing.
All right.
Yeah, not that old. Yeah, just do something.
Okay, we want to interact with the Professor Grobler, and thank you so much for the interesting presentation. And you've taken us, through a nice tour. And I want to invite us to, if you have a question or a comment, to the professor. Okay.
Yeah, thank you very much for that elaborate presentation. We have learned a lot. But this is just a discussion, really, or just an idea. You said about communicating the science from research through papers, conferences, etc., and sometimes workshops where you meet stakeholders, such as managers and so on. But I'm thinking of policy briefs which are targeted towards policy makers. So how do you look at that? Because you didn't mention them, or one at least. Thank you.
Can you hear this microphone? But there is a bit between the different research questions. So when it comes to the ones that can cause a lot of danger, like hybridization, there's fairly, well, there's a lot of legislation in place or being introduced on that. And very much in terms of moving things out of the country and mixing it with our populations, because we're going to sit with the legacy of that for, in some areas, for a long time. So there's certainly directives in terms of that. In terms of diversity, there's nothing. It's for the owner to decide.
Thank you. You okay with that response? Anyone else with a question?
Good morning. My name is Samuel Karaoke, University of Eldoret. That's a very nice tour, I would say. And I've noticed that in your diversity studies, you've used mitochondrial DNA sequencing in some of your slides. And in others, there is toll-like receptors. So is there a particular reason why toll-like receptors would be used in genetic diversity studies? Is there a specific reason?
Yes, I think in a way you can see it as a transition. Well, it's not only a transition because it's very much relevant now as well. But in the whole development of how we study diversity, from microsatellites as a neutral marker to the current genomic situation, certainly in lots of studies that I know, looking at specific genes with potentially adaptive significance was a bit of an advance of just getting the neutral data on levels of diversity. So the idea is to go beyond neutral markers and start exploring what doesn't matter if you don't have enough diversity, and the way you do it is the easy one to do it is to target something that is, for example, linked to disease resistance, and if the variation is also lost at the same time that you lose neutral diversity.
Yeah, I was thinking in the same light because toll-like receptors will be involved in innate immunity. And obviously, the exposure of the environment, what the maybe animal is experiencing, would really adapt on that at that particular level. So I think the geographical or the environment, I think toll-like receptors is a good proxy to study that. And on the COVID and vervet monkeys, you said that you noticed that monkeys that are in captivity, they are also more likely to get COVID, according to the table that you showed. And you said that you stopped because maybe COVID cases went down. But I think it's also interesting to continue studying. SARS-CoV-2 is one of them. But I think there are other maybe pandemics that are on the way. So I think it's good we have this kind of One Health approach, such that we not only protect ourselves, but we also protect our animals as well. Yeah.
I have to say there's also a totally other side to that. And I'm actually curious to know if it's the same around here. Our guest from KwaZulu-Natal will also appreciate this. Monkeys have I always say they have an image problem in South Africa, but they are really persecuted. People in general do not like, or a lot of people do not like monkeys because they can raid the campsites and they can raid suburbs. If you look in KwaZulu-Natal, and they come into your house and they do things. So in many places in the country, rabbits are not popular, and they suffer a bit in terms of the way people go after them. And when we started doing this work, actually the first thing I did was to find a senior person in KwaZulu-Natal conservation, because we decided we really have to be careful about the narrative here. If monkeys are already not particularly popular, and words comes out that and now you're also going to get COVID from them, that would just accelerate everything. So I have to say I was also a little bit cautious on how that one could be done. But yes, there's a whole different thing. They could also be used as model organisms for studying lots of aspects around disease.
Oh, thank you. Anyone else? We have 2. I want to know so that we budget time. Oh, 2? Okay. You mentioned your name?
I'm Olivia Dimov from Germany, and thank you for your presentation and for covering these vast amounts of projects. It was very interesting. I wanted to ask about the touch DNA against poaching. Are there plans to pursue this further, or was this just like a one-time project to prove it's possible? Because I imagine tranquilizing the rhinos is not very easy, and maybe it's not very applicable, but maybe there's already other plans to pursue this kind of project.
Yes. Well, we did the practical because owners handle animals a lot. They get tranquilized anyway for barking and for disease purposes. So yes, we coordinated to use those opportunities, and yes, I'm hopeful that will actually eventually be applied, so that it can be done from poached carcasses, and we're taking it further. I'm not going to mention the species because it's not published yet, but we're actually looking at a very different species as well with the same principles.
Just one follow-up question, because you could detect humans on the rhinos, but do you think it will be possible to detect like certain, like I don't know how prosecution is in South Africa, but could that be linked to like a data bank or something to find the individuals that are responsible for poaching, or is that maybe too far?
You mean the individual humans?
Yeah.
Yes, that's a topic of lots of discussion in South Africa and many other countries. There are very few countries who allow population databases of genetic profiles of everybody. So we have a database in the country, and I think most countries work similar, where you enter the database if you're arrested, and then it will stay in the database for a while. So if you happen to be a repeat offender and it's in the database, of course it can be detected, but only if you've been genotyped before.
Hello, my name is Olivier from Benin Republic, and thank you very much for your presentation. I have to mention that I'm not an animal scientist, I work on plants. And I just want to ask a very general question, which may be off topic. It's about the use of artificial intelligence in genetic conservation of animals. Just wanted to know, like, as an expert, what do you think about integrating AI in genetic conservation of animals? Thank you.
There's obviously a huge possibility for that, just like AI is exploding in terms of everything that we do. But yes, I mean, if you think back of the question or the scenario I posed right at the end, easily understandable strategies in terms of what is needed in terms of augmentation of populations to not lose diversity, or modeling of outcomes with certain frequencies and first certain sexes of introductions. Yes, if you can train your AI to do that, that could make it a lot easier to use.
Thank you so much. One last question from.
Yeah, thank you very much, Paul, for the presentation. As you made the presentation, it was coming out very clearly that genomics information is unfolding a lot from what we knew before. So my question is, how do you see this unfolding, complexing further our research and recommendations? And are we set to unwrite some of the information we previously knew? And I mean, I talk as a crop improvement lecturer and also as a researcher.
Okay, there's actually quite a bit one can say about that. I would like to think for many cases we should not think of having established answers from traditional markers, and genomics changes everything, although there are certainly cases where it does change anything. So I like to think of it more as additional information. What is interesting, and the other academics in the room may have also some views on that. We're living in a transitionary stage where it's everything is genomics now, but there's still lots of scientists out there who can do microsatellites and regular Sanger sequencing. But I think we're going to lose some of that capacity, because as a new generation of students come into the system, and with the constraints of time and funding, they're going to start off, and not going to start, they do it already. They start at the genomic level, and the other markers are not used that often anymore. But then again, a lot of the information is in a way similar. I'm very interested in diversity, and it now just gets nicer. You had 20 microsatellite markers 10 years ago, and now you have a couple of hundred SNP positions, but and it's a lot better because they're nicely distributed. But it's just a nicer way to do the same thing.
Yeah, but maybe as a follow-up, if I may, we looked at the genetics, yeah, and we looked at it as a linear relationship. Now, genomics is coming out to show us that it isn't as linear as we had anticipated. So, are we going back to tell people that, look, sorry, we've made new discoveries? Because I really don't think it is going to be just as simple as adding more information. Yeah, so that's my concern.
Yeah, that can and will happen, but I don't think it should be a concern. We can, and the nice thing about genomics is once you have the data, like I said before, it's just a start. Yeah. So you can discover or uncover mechanisms or gene regions that drove diversification that nobody even thought of before. That doesn't. Well, in some cases it can contradict the original idea, but again, mostly I would like to think of it as additional information.
Wow, thank you so much. Maybe. Yeah, thank you. Maybe we need to appreciate Professor Grobler. I don't know how. Maybe just raindrops. Is there a club like that? I think I had a few months ago, with my students saying they can do raindrops. Yeah, and I think they were doing like calling for rain. Yes, I think because Savannah we have a lot of rain now. So we can do that. Yeah. Let's start. Hi, we can't hear anything. Thank you so much.
Thank you, I really appreciate
for the
that.
nice presentation. All the questions I had has been asked, and you've done a very good presentation. We are so happy, and we've learnt a lot. Thank you. So our next program, from our program, we are heading for our tea break. As we go for tea break, our washrooms are to the right. When you exit from here, it's to the right. When you exit from there, it is to the left. And we'll have our tea from this side. So we'll, I think, use this door, or if I want to welcome again all of us. And we'll go back to the first session of our program, which was an introduction. And I'll invite James Barasa to come and do the introduction part so that we can now know each other better. And then we invite our guests. Karibu, James.
Yeah, so thank you once again. Our chief guest, the DVC, who is representing the VC, feel welcome. But before we begin other activities, may I just take this chance to once more welcome everyone, all the guests to this meeting, which is mainly interactive, and I hope that you will keep enjoying the discussions and presentations. So may I take this chance perhaps just to welcome members of the planning committee. I think they were very instrumental in all the arrangements and plannings. So maybe the planning committee, you just stand and mention your name. Mention your name and your institution.
Good morning. My name is Samuel Karaoke, University of Eldoret.
Good morning, everyone. I'm Dr. Kanya Kennedy, University of Eldoret, School of Agriculture.
Good morning, everyone. I'm Dr. Vivian Tuwei, School of Medicine, Moy University.
And I'm Emily Chemoiwa, Department of Biological Sciences, School of Science, University of Eldoret. Thank you.
Thank you, members of the planning committee. Of course, we also had the director of research, who is away on other activities, Professor Liz Mwambri. She was also in the planning committee. My name is James Barasa. I chaired the committee, and I enjoyed working with the committee members. Thank you. Yeah, so next we just request to make a general introduction of everyone so that the chief guest can have a feel of who is around and who the guests are. Yeah, so perhaps we just start with the professor. You just say your name, your institution, and also your country.
Yeah, is it working? Yeah. I'm Ralf Tiedemann, University of Potsdam in Germany, and I'm an evolutionary researcher.
Yes, I'm Paul Grobler from University of the Free State in South Africa, and focusing on conservation and management aspects of that.
Thank you. Keynote?
So we will have the keynote guests, keynote speakers first.
Yeah, I'm Miriam Kinywa from University of Eldoret, a plant breeder.
Cyprian Katongo from University of Zambia. I'm a geneticist.
Yeah, thank you. Those are the guests who will give keynote speeches. But we also had a professor from Nigeria. She was unable to travel because of other urgent activities, but she will be presenting online. She's Professor Anise Harper from Redeemers University. She was unable to travel. We also have Dr. Josiah Kuja from Walter Reed. He will be arriving tomorrow. Yeah, he will be arriving tomorrow. Thank you. So we go to other guests. We could.
Yeah, hello, my name is Michael Latov. I'm from South Africa, University of KwaZulu-Natal, and I'm doing research on honeybees and pollination.
My name is John Kiplagat, PhD student, University of Eldoret.
I'm Dr. Hilda Japleting, graduate of University of Eldoret.
Good morning. I'm Anne Legong, a master's student at the Cambridge Graduate School, currently based at the Walter Reed Labs here in Kenya.
Good morning. My name is Waswa Sarah, an MSc student at the University of Eldoret.
Good morning, everyone. My name is Daisy Simeo, an MSc student at the University of Eldoret.
My name is Purity Muhongo from Barara University, an MSc student.
Good morning, everyone. My name is Catherine Silali from the University of Eldoret, and I'm an undergraduate taking microbiology.
Hi, everyone. My name is Olivier Dayo, originally from Benin Republic in West Africa, but I'm doing my PhD in Germany at the University of Munster. And my research focuses on the interaction between parasitic weed and host plant. Thank you.
Hi, I'm Olivia Dimov, PhD student and veterinarian from the Helmholtz Institute for One Health in Germany.
I am Sharon Nauma from the Department of Chemistry and Biochemistry from University of Eldoret.
Good morning, everyone. Blashu Sereko Bambal is my name, from Uganda, and I'm a PhD student from Busitema University. I am working on mosquitoes as herbivirus vectors. Thank you so much.
Thank you. So we might give a chance to other technical team, members of the technical team, starting with.
My name is Peter Bahati, ICT department in the University of Eldoret.
Good morning, everyone. My name is Victor Kimutai, Corporate Affairs and Marketing Team, University of Eldoret.
My name is Samuel Namnaba, providing media support. Thank you.
Thank you, everyone.
Thank you so much. I'm Louis Mabonga. I work in the office of the DVC, Planning, Research and Extension, University of Eldoret. Thank you so much.
Hey, good morning. My name is Lamek Josh Menya Otieno. I work in the office of Planning, Research and Extension Division of the university. I'm also a postgraduate student in the Department of Fisheries and Aquatic Sciences. And in this conference, I am part of the secretariat. Thank you so much.
Thank you. Sylvia, just mention your name from there.
Okay, thank you, everyone. That gives you the DVC, our chief guest, some general idea of who is in the room and some of the expected presentations. Yeah, so without talking much, I will take this chance just to appreciate the founder, the Society for Molecular Biology and Evolution. They have funded everything. including air tickets for the guests and the general costs. As you can see, we've even made the book of abstracts, the program and everything. It's courtesy of SMBE, Society for Molecular Biology and Evolution. This is a very good society that supports molecular sciences, and annually it supports small meetings across regions. And so we were fortunate to get the support to hold the Africa region meeting from here in Eldoret. There will be other regional meetings in Europe, even the America, and I think Asia. Professor Tiedman is aware of this. So we are very fortunate to get the support to host the Africa regional meeting. We thank them for that support. Then secondly, we wish also to thank the University of Eldoret for facilitating all the activities, all the acquisitions, and the support they provided. So Bwana DVC, when you go back and meet the VC, pass our appreciation and just let him know that we'll continue relying on his support. So we are very grateful that you have also come, and if your time allows, you are free to interact with us, interact with the guests, and listen to the presentations even the whole day. So let me stop at that and pass over the microphone or the chance to the MC. Thank you. So on that note, may I invite Professor Miriam Quinyo, who is a professor in the School of Agriculture, to welcome the DVC to make his remarks. Professor Quinyo.
So thanks, James. It has fallen my lot. I was coming to the conference, and now I have had the pleasure of sitting next to the acting VC. You know, these things come a few times in your lifetime, so it's good to make use of them when they happen. Like I introduced myself, I'm Miriam Kinywa, and at the School of Agriculture, University of Eldoret. And my singular role is to describe or introduce the acting Vice Chancellor, and also to invite him to open the conference. I can easily talk about our DVC, academics, and student affairs, Professor Netondo, because he makes sure that we work together by bullying us into getting to his programs and helping out, and that way we get to interact. So Prof, we are happy to contribute to the academic affairs and the student affairs at the university. Professor Netondo is a physiologist and biochemist. He is the Deputy Vice Chancellor at the University of Eldoret, in charge of academics and student affairs. Academic matters at the university include conferences. That's why he's very well fitting to be here today on behalf of the VC to open this conference. We are fortunate enough to have a scientist who's related on the line that we are discussing to open this conference. Prof, welcome, and we are happy. as the people have introduced themselves, you've been able to hear that they are related to the subject of today. This is a conference for the molecular biology and evolution, SMBE. That's what SMBE stands for. And for today, we are participating in a regional symposium on evolutionary genomics in agriculture, environment, biodiversity conservation, and health for Africa. We are trying to put our minds together to see what can come out of genomics for Africa today. We have key speakers, keynote speakers, and of course other presentations. And we also have plenary discussions, so that at the end of the day, it is a 3-day workshop, not workshop, conference. Yeah, a 3-day conference, and we hope by the time that we are leaving here, we'll have come up with some of the suggested solutions for Africa as far as genomics is concerned. So without much ado, I wish to invite you, Professor Netondo, to please come and open the workshop. Please let's clap for him as he comes.
Thank you, Professor Miriam Kinyua, for the nice introduction. I've just listened to the introduction of the participants of this symposium, and I realized that it is cutting across continents. From Germany, there's Europe, we have Africa, and I think some, is it from Asia? But it's a good mix of researchers that are participating in this symposium. Before I read the speech of the Vice Chancellor, I just want to say that I'm also a biologist, as Professor Miriam has said, a plant physiologist of long standing, over 30 years in the university. I have been teaching at Maseno University. Those who know where Maseno is, it's near Lake Victoria. And I'm proud that one of my students is here, Dr. Chemoyo. Thank you so much. So, because we have people from Germany, from Uganda, from South Africa, I can say good morning for the Germans. Karibu for the Kenyans and Ugandans, and South Africans. Good morning to everybody. Yes, I have the speech from the Vice Chancellor. Apparently, he was supposed to come very early in the morning, but because of some other emergencies that cropped up, he could not make it, and therefore he asked me to come and read his speech and open this symposium. Therefore, I just want to go straight to the speech. Distinguished guests, representatives of the Society for Molecular Biology and Evolution, SMBE, esteemed scientists and researchers, members of faculty, postgraduate students, ladies and gentlemen, good morning. It gives me great pleasure to welcome you all to Eldoret and to the University of Eldoret for the SMBE Regional Symposium on Evolutionary Genomics for Africa 2026. With the generous support of the Society for Molecular Biology and Evolution, we are honored to host this important symposium that has brought together eminent scientists, researchers, scholars, and students from different parts of Africa and across the world. Your presence here reflects the growing importance of collaborative scientific inquiry in addressing global and regional challenges through genomics and evolutionary biology. This symposium provides a valuable forum for strengthening collaborations among scientists on the African continent and colleagues from the global north. It also creates meaningful opportunities for postgraduate students and early career researchers to connect with the experts in genomics from diverse countries and institutions. Such interactions are essential for knowledge exchange, mentorship, innovation, and the advancement of scientific research. As the University of Eldoret, we are delighted to have been accorded the opportunity and support to host this regional symposium for Africa here in Eldoret. We sincerely appreciate SMBE for its confidence in our institution and for supporting initiatives that promote scientific excellence and research capacity development on the continent. Beyond building collaborations and professional networks, this symposium offers us an excellent opportunity to showcase and promote research in molecular sciences at our university. We are particularly proud of our growing research infrastructure, including the Fish Genomics Research Laboratory and the Biotechnology Center, which continue to contribute significantly to scientific research, innovation, and postgraduate training in genomics and related disciplines. I've had the opportunity to look at the symposium themes and sub-themes, and I must commend the organizers for sampling a rich and diverse scientific program. Of particular significance is the strong participation in research focusing on antimicrobial resistance, commonly known as AMR. This remains a major global health concern that requires robust scientific data and evidence-based interventions to support governments and health authorities in designing practical, sustainable solutions. It is our hope that over the next 3 days, you will engage in fruitful deliberations, productive discussions, and meaningful exchanges that will strengthen partnerships and inspire more active collaborative research initiatives. We also hope that this symposium will open up additional postgraduate training opportunities and research networks in the field of genomics and molecular biology. As you participate in this symposium, I encourage you also to experience the warmth and hospitality of the people of Eldoret and Kenya at large. On this note, allow me to once again warmly welcome you all to Eldoret, Kenya, and wish you successful deliberations and a pleasant stay. It is now my singular honor and privilege to declare this SMBE Regional Symposium on Evolutionary Genomics for Africa 2026 officially open. Thank you very much.
Yes, that was my lecturer when I was starting off as a very young girl at the university, Maseno University. And I'm so proud that from his teaching in my first year all the way, I'm able to stand and say I'm a very good researcher. So thank you so much, Prof. We appreciate our Vice Chancellor for being here and for opening this symposium. Earlier on, Buona VC, we had our first keynote speaker, Professor Grobler, who gave us an insight into African conservation, genetics, and genomics. And he took us through some areas that he's an expert in, and we were so much inspired. One of the things he talked about is that there are possible collaboration even in doing research, including looking at the porcupine and comparing what is in South Africa and what we have in Kenya. He talked about many other things in terms of conservation of the wildlife. And so that was our first keynote speaker. And I must say, you know Professor Romulus Abila from Maseno University. He has worked really closely in research with James and Professor Grobler. Prof. Grobler, I don't know whether you remember Professor Abila, James Abila. Yes, Romulus, yes. So he was my supervisor for PhD as well. So Maseno has really mentored many. So welcome again, and our next keynote speaker is Professor Dr. Ralph Tiedemann. And a while ago, last year, we interacted so much with Prof. He came to our university, our lab, and he trained us on bioinformatics and also on molecular lab. We had wet lab demonstrations from his lab in Germany, and we were so happy. He studied biology and computer science from Kiel, German, and Reykjavik, yes, you'll forgive in Iceland. He has a PhD on bird genetics, associate professor in Kiel. He is a full professor in evolutionary biology, University of Potsdam, since 2002. He's a guest researcher at Brussels and Oklahoma, and his topic today is on evolutionary genetics, ecological speciation and adaptation, conservation genetics. Yes, yes, he has done research mainly in fish and birds and mammals, and we are so happy to have you, Prof, talk to us. Karibu sana. Welcome. Clap for him as he starts off.
Thank you, Emily, for the very kind introduction, and thank you, the organizers, most prominently James Barassa, for inviting me here. It's really a privilege. Before I start to speak about our research on electric fish, I'd like to expand a bit on my background. And I'm coming from a city in Germany, Neumünster. It's very small. You may not know it. It's north of Hamburg. Most people don't know Neumünster. And my point I want to make is that science for many, at least for me, is kind of a combination of chance and taking the chances. How that? You know, when I started as a birder, because my dad was also a birder, so I loved birds and I want to do everything about birds. And originally I was concentrated more to the north, so I went to Iceland, very cold city, but also very expensive city, so country. So I needed money. And so I hired in a population genetics lab, and that's how I became a geneticist. I was actually there as a birder, then I worked with Drosophila, and then I thought, why not applying this for birds, and later for a lot of other species. And that is actually one aspect of chance. The second is I had done some research in East Africa, mostly Kenya and Ethiopia, for a couple of years. And then about 6 years ago, I received an email by a researcher from a city I had never heard the name of. That was Dr. James Barassa from Eldoret. And he had the idea to make a joint application on tilapia, which we proposed to the German Science Foundation, and we were successful. And that was the spur of I think, very fruitful and also delightful collaboration. So that was, again, chance, but taking the chance and making very good thing of it. And the third is actually the topic of today, that is this fish. As I said, I come from a bird and mammal background, but I got involved as a reviewer in a project on sturgeons, these ancient fish. And that very researcher, Professor Kirschbaum from Humboldt University, over a beer after the sessions, he said, I'm also working on electric fish. And I was completely fascinated by that. And I also keep them, he told me. So I went to Humboldt and I saw the fish. And since then, that is about 24 years ago, this has become one of our major topics. And again, it's by chance, by random, this opportunity appeared. But I hope I have made the best out of it. This is actually a long-term project in the sense it involves many, many people. Apart from Professor Kirschbaum, it is most notably Professor Victor Mamonekena from University of Brazzaville in Republic of Congo. Those of you who had the chance to be on the last PAFA conference in Brazzaville, that was organized by him, because these fish originated from River Congo. And then it's a number of, most people you see here are former PhD students, so it's a series of PhDs, so always new questions arise, and then we hire new people and then try to address these issues. So what is this electric fish about? I mean, the electric sense is something we really don't have. I mean, you may know, there's the electric eel in South America, and that is a strong electric fish. So that is so strong that we would also feel it. It's even dangerous for us. It's 500 volt. But with these weakly electric fish, they discharge very minor voltages. It's about 1 volt. You can actually not feel it. You can measure it, but you cannot feel it. And interestingly, in Congo, the fishermen who fish these fish and they sell them or eat them, they don't know they are electric. So that's because you cannot feel it. This sense has evolved several times in vertebrates, 6 times alone in fish. And the most prominent 2 lines are the unrelated South American gymnotiforms, also known as knife fishes, where the electric eel belongs, and the African mormyroids, which we mostly work on. And the important about these electric fish is not only are they able to sense electricity, low voltages, but they're also able to produce it themselves. So they're actively producing an electric field, which they use for location. So in this electric field, they produce, if you have it distorted by an object, they can sense it. And the River Congo, at the area we are working, that's between the cities of Brazzaville and Kinshasa. It's very, very turbid. So there is these rapids. It's really a turbid river. So sight is not very helpful. So they use that for orientation. They also use that for communication. So they can discern between conspecifics and heterospecifics, but they also find their mates by these signals. How do they do that? Well, they have specialized muscle cells in their caudal peduncle, which are called electric cells, and these cells have lost the ability for contraction, but instead gained the ability to produce an electric signal in a highly synchronized way. It is they are able to excite all these cells together to produce a signal of the length of microseconds. So it's highly synchronized, which is very interesting in its own right. One aspect of interest here also with regard to genomics is that the teleosts in general have undergone a whole genome duplication. So every gene we have once, they have twice. And that has raised the opportunity for them to actually have one set of genes evolving towards serving the electric sense, while the other retains the original important function for the muscles. Within the Marmoraids, it is actually a rather basal group of fish which is reasonably species poor, except for the Marmoraids. There are very many species. So that gives us the idea that this electric sense is really a key adaptation, enabling them to diversify into many species. And if you look, the basic split, it's into two groups, one of which only consists one species and the other many species. And we still find new species there. And the major difference between the two is the one has an electric signal, EOD, I should say, it's jargon, it means electric organ discharge, which is wave type, and we are working on these which are pulse type. More specifically, we work on a specific group of these elephant fish, as they are also sometimes called because of their long trunk. And depending on the authors you ask, there are 3 to 16 described species. What you see here depicted is the general distribution by river systems, but most of the species we focus on, all but 2, all live in the Congo River. It's a huge river system, of course, but again, a chance item, we can say all our work is more, say, sympatric for logistic reasons. So all of our work was carried out in the surrounding of Brazzaville, Kinshasa. So it's admittedly a very minor fraction of this huge river system, but that's the current status. We see here these discharges. You see different phenotypes. You see they differ by the length of their snout, also the height, but most prominently they differ by the length of their discharge. And you see here a very short discharger. It's about a microsecond or 10 microseconds, and these go up to 200 milliseconds. So it spans several orders of magnitude, and we are specifically interested in understanding why that is, both from the perspective of how is this made, but also what does it mean for the ecology of these fish. Fieldwork in Brazzaville is actually much more easy and less adventurous than you would think. You just stay at a hotel in Brazzaville, you take a cab to the seaside, and then you negotiate with fishermen who fish the fish for you, and you just buy them from them, and you have them. And then you go to the university in Brazzaville, and you can take your measurements. So it was perhaps more easygoing than it sounds in the first place. And you see here the area of the rapids, which is really a very turbid area where these fish occur. A first question was, how many species are there? So we find the more you look, the more species you find. You see, okay, some have clear names, but then we found within one species, we all of a sudden realized, okay, there is one has this strange dip first in the EOD, the other not. Is it perhaps also 2 species? And we also found a few which still don't have a name. We already found them some years ago. We always think we have to describe them, but we don't have names on them so far. And that is a rather recent tree from a former PhD student, Francesco Lamanna, where you see a general relationship between these fish with a basal group called Tamandua, and then we have here this group and the second group, which we always call the Alces group, which is not very well resolved. And importantly for us, we have actually some of these fish alive in our lab. And these originate from 2 sources. One is to bring them alive from Brazzaville, which is actually more of a challenge than getting them, and mostly for how to say, for really the regulations of the European Union, bringing fish in is really an asset. Even you have all the papers, it's really difficult. And the second is getting it through a wholesaler in Frankfurt, which actually imports tropical fish, because some of them are also used by amateurs for aquaristic. And a follow-up PhD student, Julia Karnitz, she actually established via DDRAT, so that's the relation to genomics. She established with DDRAT over 1,000 SNP loci. We already heard from Paul. SNPs are these single nucleotide polymorphisms, which in singularity don't carry too much information, but you have many of them, you can get very, very high resolution. And what we actually find, all our species are clearly separated, clearly valid species, except for this, what we call the Alces group, which is a huge of a turmoil. A side effect of that is we give all these fish, of course, names. And we think that's the best of it. Then you all of a sudden may realize, but is that really the right name you gave these fish? It may be consistent within your own research, but other researchers use the same names perhaps for something different. We're currently realizing there's a US study also working on electric fish, and the names do not really match, and we try to find out why that is, because it is not trivial. But the type material is to a large extent in musea in Tervuren, Belgium, and Paris, but they are all in formalin, so they are not easily not easy to retrieve DNA from that. And another aspect is, and this is a particular representation of a tree where you see in blue the most supported tree, but you see in other colors alternatively supported trees. And you see that in some areas of the tree, actually the genomes don't tell a single story. Some parts of the genome support one tree, and other parts of the genome support another tree. So the idea of we have the whole genome, now we have the whole truth is not as simple as it may seem in the first place. On top of that, with a so-called tree view analysis, we could also infer that there is essentially ancient hybridization. So you see here an introgression event, you see another introgression event. That complicates again. It also relates to a topic already raised by Paul, that there is hybridization also out in nature. It may be infrequent, but the more data you get, the more you find the signs of this, signs of this introgression in the genomes. Okay, so what the general outset of our research is what causes this divergence and the speciation, and we tackle it from a proximate perspective, what are the anatomical physiological mechanisms, and from an ultimate perspective, what is the adaptive value. Just focus first on the first one, and I should say there's a German saying which says, if you only have a hammer, every problem looks like a nail. And our hammer is genetics, so we always look at through the lens of genetics. Other people, they go to the physiology or the ultrastructure of the organ, which is also important. And we're also reasonably ignorant towards the receiver side. We only work on the electric organ, which is the sender. But evidently the fish, to make use of this signal, needs also the receiver side. But that will not be covered today. And one aspect we immediately realized is that there is this difference in snout morphology, and we quantify it with geographic morphometrics, and could actually, with landmark analysis, show that also by the morphology, you find also these species. You cannot, I mean, if you see them, it's difficult to really tell them apart, but if you make a quantitative approach, then you, by PCA, you find the different clusters. Another aspect is, if you go into the ultrastructure of the organs, you may find these are the electrocytes. They are piled up as in a row, and they are innervated either from the posterior or from the anterior, and that difference decides whether the polarity of the first phase is negative or positive. So that is a relation to ultrastructure, which has to be considered as well. So we see here a further is this whether you have stalks which go through the cell or not. Stalks are these innervation patterns, and if they go through the electrocyte, you get this first dip, and if they don't, like in this species, you don't get this. So you have a very, very similar discharge, and the only difference, this one has a small dip in the beginning, this one hasn't. And these fish, it's in the realm of microseconds, they can distinguish between them. Sorry. A further aspect is, and we thought then what could be evident candidate genes to look at. And we thought, well, electricity, perhaps ion channels. So we looked at the expression of ion channels in the electric organ relative to the skeletal muscle. And our idea was those genes should be of importance for our signal, which are specifically upregulated in the electric organ relative to the skeletal muscle. And you see this representation here, 0 here. Okay, now that's the one thing. So we found out those genes. And the second was that we said, okay, let's see one fish which has a long discharge, another which has a short discharge, and we show which genes are more expressed. The general pattern is the long dischargers, they have a higher expression level of many ion channel genes. So we kind of think this relates to their ability to produce a longer signal. We put it then to the entire genome scale, and that has been promoted further by my current postdoc, Feng, and he will have a separate presentation on that later today. But to give you a small glimpse on that, once you have the entire genome, you can actually also, you can see expression level differences, but you can also see, are the transcripts different? You can mine the transcripts for SNPs. And that's what we did. Under the idea, the expressed genes are those which should matter for the organ. If we find expressed differences in them, they may be informative. So that is, again, Julia Karnitz. She developed a pipeline to do this for these two species, a long discharger Chokwe and a short discharger Compressor rostris. And from about several thousand genes expressed, she identified 201 genes specifically expressed in Chokwe, with a long discharger, and 192 in the short discharger Compressor rostris. And this is perhaps a bit difficult presentation, but I want to guide you through it. You have in red here the Chokwe and the long discharger, in blue the short discharger, and you have two bars. One is the representation in the entire transcriptome, and the second in those which are enriched for different SNPs. And what is important is if you find a difference here, if it's this direction, it means this particular category of gene ontology, metabolism, is specifically enriched in those expressed in the electric organ. And another one, which is this organismal systems, it's particularly or significantly less abundant in the specific genes as compared to the entirety. And by this, we identified quite a number of really interesting candidate genes to be further investigated. And again, this representation shows you randomness in the gray zone, and you see what is upregulated in the long discharger processes here, transcription or cancer genes. Cancer is, of course, typically not a very favorable state, but cancer genes can be often related to functional change because they change the cell processes and may be of interest in this context. On the technological side, we have a very talented animal keeper, Lin Nguyen, and he he managed to not only to breed these fish, but also to crossbreed them. And that means we actually are in the position to be able, what happens rarely in nature, hybridization, we are able to do that in the lab. And that offers quite some opportunities for this type of analysis, because these hybrid fish, they then have two sets of genomes from the respective parent, and you can actually see how gene action then interferes with the signal divergence. And that is recent work of Feng Sheng. He has these hybrids, and then he asked himself, is the expression of genes related to the length of the discharge? We have here different length categories. You see 5 milliseconds, 0.4 milliseconds, 40 milliseconds. And on top of that, in the hybrids, he was interested to see are the different alleles coming from the two different parents differentially expressed. The first which he found was really interesting in this, you see in this principal component analysis, it's kind of stratifying the entire genome according to the two first principal components. And on the first principal component, you find the distinction between the skeletal muscle and the electric organ. So the electric organ, although it originates from skeletal muscle, is completely different. But then, on the second axis, you have here the short discharger, here the medium discharges, and you have here the long discharges, meaning that there is a systematic pattern of differential expression relative to the length of the signal. Feng then classified these genes by categories as to their pattern, and we were mostly interested in those which were either upregulated with increasing length of the signal or downregulated with increasing length. And by that he identified a number of really interesting candidate genes, among which this particular gene KCNJ2, which is an inwardly rectifying potassium channel. And what he then could verify in these hybrids, that actually the two alleles, the black part comes from the short discharger, and the pale part comes from the long discharger. There's allele-specific expression. So they have much more expression of those genes which actually make a long signal. The next step now is, and Feng has already progressed on that, is to try a knockout and see whether really this effect is related. He has already succeeded doing that in the embryos, but that does not give us the adult phenotype. As of now, our major obstacle is in Germany, it's not easy to get permission for animal experiments, so we are currently seeking for the permission to do what we are already able to do. Let's see how that goes. A further aspect is that in the ontogeny of these fish, you have very early on a larval signal, and then at a certain stage, all these species have the same length of discharge. And then in one species, this stays the same in the adult, and in others it goes through several or even many different steps to the permanent adult, much longer signal. And again, from our perspective as geneticists, we always thought, well, in this one nothing happens because it stays the same. So we look for the underlying reasons why it's elongated in the others. Until we spoke to a physiologist, and he said, well, actually the organ is growing all the time. So actually the signal should change. So maintaining it the same is an active process rather than nothing is happening. So now our focus turns a bit around. We have to understand how these can actually keep the signal constant while the electrocytes are constantly growing. And that again shows it's really important to seek expertise from different fields. What we also see as soon as they start producing the longer, they get these perturbations, which we call papillae in the electrocytes. So there is a thought that this histological aspect relates to the length of the signal. Okay, what our future plans with these hybrids are understanding how really the ontogeny works of this, try to produce F2 for co-segregation, and make a better assessment of the histology of the hybrids. But all what I told you so far is just the proximate side. It's just working on how the signal is produced. But wherefore, you can ask. What is the reason? Why are the fish doing this? So is there any adaptive value of the divergent morphology, or is there adaptive value of the divergent EODs? What is the role of the EOD in total in this radiation? So we said we all the time work at the same place, but it's if you look closer in Brazzaville, the habitat is actually not always the same. So you have these rapids where it's very, very turbid, but if you go a bit upstream, it becomes very calm and sandy. This is a rocky habitat, huge rocks. This is a sandy habitat. And if we look closer, we see some of the fish, they are more in the sandy habitat, some are more in the rocky habitat. So, so then we had the idea, perhaps these different snouts are adaptation to different substrates. So our naive thought was, if you have a long trunk, you can go between stones, and which, if you are feeding on sand, you don't need that. That's another work from an Egyptian PhD student of mine, former PhD student, now postdoc elsewhere. And Rahma made a very simple experiment. So she had this choice tank, and she had two substrates, and she actually offered them insect larvae in these trays, and just observed, or let a video camera observe what the fish are doing. They are nocturnal, so it's at nighttime, but you can illuminate it with infrared, and if you have an infrared camera, you can actually capture. And what she found, this is a heat map of their presence. And we had actually 3 species with different snout lengths. And this one is with a long snout, this one with an intermediate, and this one with a short. And here on the left is the rock, and on the right is the sand. And you clearly see the long-snouted clearly prefers the rocks, while the short-snouted clearly prefers the sand, and the intermediate-snouted is actually indiscriminate. So we can actually, we have observational behavioral evidence that these differences in morphology relate to feeding behavior. And this is the quantitative aspect of it. So she could actually show that the short-snouted clearly prefers the sand, the long-snouted the rocks, and the intermediate-snouted intermediate. She also looked how often are they probing, because being in that area is one thing, another are you really trying to feed there, and the same pattern holds also true for the probing incidences. Another aspect is then, what do they really feed on? Nobody knew what they feed on. So we actually made a DNA metabarcoding. That was also Rahma who did that on the gut content. So while we were in Brazzaville, we caught a lot of fish, or I should say the fishermen caught the fish. We bought them from the fishermen, and also took stomach samples. We tried to use ordinary metabarcoding, but that didn't work very well, and we suspect that they had a quite active intestine system, so the degradation was already too high. So what we then did is actually a bit of a sophisticated method. So we used so-called targeted hybrid capture. We produced particular baits, is it called? That's actually kind of fishing units for particular DNA parts. And so we fished then DNA out of that. And to make a long story short, then you get a representation of what a particular fish is eating. And this is just an example. It's a so-called Kraken analysis. You see all what they eat is eukaryotes. Okay, perhaps not surprising, but most of it is arthropods in this particular species. Most of it arthropods. And then if you go closer into, you will find a lot of it is actually mosquito larvae, which was kind of thought anyway they would eat. But you see also some others. There's some worms and mollusks they eat. And then Rahma compared that systematically across the different snout lengths and discharges, and indeed could establish that that there is a different food spectrum between the long-snouted and the short-snouted, and also between the long dischargers and the short dischargers. Again, we don't know whether it's a preference or whether it comes just that in the different habitats there are different prey items available. So as a bottom line, the short-snouted mostly feed on mosquito, while in the longer snouted, the longer discharges, there's also a lot of coleoptera beetle larvae. And a further aspect is actually the mating behavior. What we also observed is that those species who live closely together, they have very, very different discharge. And they all live in the same area, and they diverge very much. While another species which lives apart, this tamandua, which lives also here in West Africa, they have a much less distinctive discharge. And we made actually a test as to whether that really influences mate choice. We were so lucky that Paul already mentioned outreach. This was actually covered by the New York Times, and they called it Electrifying Love, and this I borrowed this cartoon from them. So we made a mate choice preference test, very simple. So we had this aquarium, we had here a female and here the males. Then they discharged, and we recorded what the fish are doing. You can, of course, say, yeah, but who knows whether it's a discharge? Could also be chemical cues. We just think it's a discharge. So we replaced the males by playback. So we constructed a device which makes actually an electric signal playback into the tanks, and the preference stays the same. So what we essentially found is when the discharge difference is very large, then they always prefer the conspecific over the heterospecific. However, if the discharge level is small, then they cannot distinguish. And then we thought, well, we now had the female as choosing, because very often in sexual selection you think it's the female who chooses. But in fish, that's perhaps not true, because they don't copulate. They kind of associate with each other. The female is laying the eggs, so the male has to find the right signal. So we repeated the experiment, everything else equal, but now the male is a chooser and the female not. And under these conditions, the Compressirostrus male could actually already also find these very, very tiny differences, which are only the first dip, so they can actually distinguish between them. What we also investigated is, because they are pulse dischargers, the order by which they discharge may also carry information. And so we tried different types, also randomized it, but then they immediately realized that's not a real fish. So a randomized, they would not react at all. So the train also covers information. So we confirm from that it seems that species discrimination and mate choice seem to be male-driven, which is not so common in animals, as far as I know. However, from one species, the males fail to discriminate, and that coincidentally, that is the only species which has a bit of a different color pattern. So perhaps there are other cues in this species. To sum up, we find an EOD divergence. The EOD divergence has a heritable genetic basis. genomic, we found that it seems to be a reproductive isolation mechanism, and there's ecological differentiation among sympatric species. So you may recall from my title, I said of a magic trait, but I have not yet said what a magic trait is. You may already know. A magic trait, well, the issue from an evolutionary perspective is if you have an ecological speciation in sympatry, they're all in this area, Kinshasa, Brazzaville, and they have a Q. But if the Q is not linked to the trait which is adaptive, then it's difficult to explain how it could evolve. So if, say, the snout is the adaptive value and the discharge is a Q, but why should they be related? So a magic trait would be if it's the same trait. So if the same trait makes the adaptation and the discharge. So that would mean that the EOD difference as such has already an adaptive value and at the same time serves as a reproductive isolation mechanism. And we are not yet there, but we still think it could be a magic trait. And with that, I'm at the end. This is night fishing at Congo River in Brazzaville, and I thank you for your attention, and I'm happy to take questions.
Yeah, I think he deserves more clap than that. Let's do a round of applause again. Yeah, thank you so much. Prof. Ralph, I don't know whether you want to sit or you want, we have a few maybe comments or questions. Okay, so I welcome all of us. Anyone who has a comment or a question in line with the presentation. So when I give you a mic, you state your name and area. You just mention your area and you ask your question.
Good morning again. My name is Samuel Karaoke, Department of Biological Sciences. I am a virologist by training and immunology. Right now my project focuses on immunogenetics, human immunogenetics. So my question is, that breed of fish, does it only stay in where there are rapids, or you can find it in other places along the Congo River or along the river, or is adapted for that particular region?
Yeah, that's a very, very good question. To start with, it's a genus which has, we think, perhaps 16 species. And the answer to your question would be different for any of these species. We always think to put them into one shelf, but any species has its own trajectory. So there are some species which can be found only in the Congo. Well, one should say the general distribution throughout the Congo basin is not fully known because not all areas are well sampled. Although, in particular, the Belgians, they have done surprisingly much in DRC. But this differs among species. And some are actually also in West Africa. And again, they are called the same name, but unfortunately, as of now, we don't have samples from West Africa. So one would have to show whether they are really the same species or whether they just look the same and the morphologists could not tell them apart. So that is, in parts, an open question. And my dream project would be to go to Kisangani in DRC and sample there, but the logistics are challenging, I was told. Perhaps, and perhaps not the safest place for people of my skin color, I don't know. But we still have that on our list to work on that question, but it's an open question. When you ask for the wider marmorites, they have a wider distribution. When I was with James at Lake Victoria, they had actually a poster there of all the fish of the areas, and they were also marmorites. And the local fishermen were not aware they're electric fish, but they're also there. It's not this genus, but the group is there. Does that answer your question sufficiently? Yeah, thank you.
Yeah, so this is just a comment, Prof, and it's very closely related to Samuel's. Yeah, I think it would be interesting if you say that the electric fish is also in West Africa. It would be interesting to see how it behaves, how this trait behaves. Yeah, because the Congo River is looking at its size, turbidity, and the mass of the water, the volume. Yeah, the discharge per minute is enormous. And I think that how would it compare with a smaller river? Yeah, that's just a comment and it's related to him.
Yeah, I would be very interested in that, and I think I already told you, James, that I have a contact to Guinea and to a researcher from there, Samba Diallo, and they found one of the species there in their collections. But they don't have the specimens any longer. They just did monitoring, and in their species list they are. But we are planning to hopefully retrieve fish from Guinea also and have a look into them. And then we could perhaps work towards an answer of that question.
Thank you. Anyone else?
Thank you, Prof, for that nice presentation. It has reminded me of my undergraduate when we were looking at animal behavior. And this aspect of EOD, the signaling in fish, sometimes signals something. I know this is an ongoing project. Could the different levels of signaling, this electric, related probably to certain fish behavior, whether it is breeding, whether it is environmental challenges, whether it is pollution, just could there be any relation to this kind of signal that is given by fish?
Yeah, again, a very, very, very good question. So in this species, the individual signal is absolutely fixed. So there's no relation from the individual signal. But the order by which they discharge is highly variable and carries indeed information of that type you mentioned. So they context dependent, make very many pulses or fewer, and they in that way communicate. And there is a group also in Germany, they had actually made a robot electric fish where they can modulate this and respond to the discharge of other fish. And then you can actually see what we also observe, because in our playback, we call that a train. We use a natural order of signaling of a certain length, say 1 minute. But then we use always the same, and you can see how the fish become bored. It's like you would meet a roboter and it says a sentence, but it says always the same sentence, and then you realize that's not a real fish, it's not a real human, it's a robot. Same here, the fish seem to feel after some time there's not this variation, which is typical. And there is indication that, in particular with regard to behavior, that the signal order or the distance between the signals carries such information.
Thank you.
Yeah, thank you, Raf, for the cool science you have just shown us. I just have 2 general questions and 1 specific one. The first one is, you mentioned the fish is present in West Africa, and you mentioned Guinea, right? So which other country can you find this species?
We know it's in Nigeria as well, and I'm not so much into the geography of West Africa. Sorry if I'm struggling.
No problem.
So I guess then the countries between Guinea and Nigeria would be evident candidates as well. It has been also found in Chad, so there are a few places where, but it's particular 2 species which are, there's one which is never found in Congo, only in West Africa, and there's another which has been reported from both areas, but nobody knows whether it's really the same species, because in between Cameroon or this, there are no records to the best of my knowledge. But yeah.
Okay, thank you. My second question is about the mechanism. So, do you know whether this electrical discharge could be involved in some kind of defense mechanism against predators, for example?
You mean it was exploited by predators?
Yeah.
Yeah, that's again a very good question. And that's something I didn't learn before the Puffer conference in Brazzaville, because I had not thought of it. But then some people said that keeping the signal short may actively avoid predation by catfish, because catfish are, I think they are not actively electric, but they are passively electric. They can sense it and they use it for predation. So in areas where you have a lot of catfish, there may be a selective pressure to keep the signal short, which is then, well, you can transform these signals into a so-called power spectrum, and the power spectrum which the catfish actually uses to sense prey is well known, and you can show that these short discharges, they avoid that power spectrum.
Cool, thank you. And my last question is about the KCNGE candidate gene that is involved in elongation of the EOD. I was just wondering how you want to go about the knocking down of this gene, and just in case it doesn't work, what is your next plan to investigate the function of this gene further? Thank you.
Yeah, good question. We are at the beginning of this, and perhaps Feng, my postdoc who will present later, is better to answer. But what I can say is we have teamed up with our physiologist, and he uses zebrafish as a model, and he has established CRISPR-Cas knockout in zebrafish, and we have succeeded in excising a part of this gene, so we can verify from the embryo that where we have applied the CRISPR-Cas, this part is cut out. So the CRISPR part works, but what we would need is, of course, the adult EOD phenotype, and for that we need additional permissions. So we could not perform that so far. But yeah, that is the way we try to do it. The outcome is unpredictable. Perhaps it has no effect, and we have to think of an alternative model. But as of now, we don't know. But the candidate, this KCNJ2, always pops up in all analysis again. So, and also this differential expression between the short and the long discharges makes it, in our opinion, a good candidate. We also find in this gene expressed mutations, which of course, again, an indication that may matter. But thanks for the question.
Thank you very much. Any more questions?
Thank you for the presentation. A very practical question. We cannot feel the discharge. Fishermen cannot feel it. So where did it start? Who first discovered it, and why did they discover it?
Well, it's now I'm going into informed guessing. I think it may even be Cuvier, or some people very long ago realized for some reason that they are electric, but perhaps I'm mixing it up, but it's at least known for a longer time. There was at times even, I think in France, the idea to utilize this sense. They had the idea to let them swim through pipelines and then measure, but that apparently never worked. But once you know it, you can of course measure it. And a very simple way of making it appreciable is you just take we also did that in the field. You take a small speaker and just have open cables and just put it into the water, and then you hear, and that's how we also showed the fishermen. We put it into their bucket and then you hear that sound. And you can also do that in the field. When we were in Brazzaville, there were people, you may have seen that, James, they had this speaker with them, and they were actually going to the river and checking whether there were fish around. So yeah, but that does not really answer your question, who found it first. I'm sure I would be quicker with Google than from my mind to find it out, likely.
Indeed, that's interesting. I know Feng will be discussing more on the same in the afternoon at 2. Anyone else with a question from the back? Thank you so much. I think we can clap for him, an organized clap again. I don't know which one. I don't know. Sharon, which clap? Sharon, you've not spoken today. Let's say clap for Prof.
Let's have a 3 factorial. 1, 2, 3, 1, 2, then 1. Let's start. Thank you, everyone.
Thank you very much. So that is our presentation of our keynote speaker in the mid-morning session. And I want to thank all of you for being patient and listening and interacting. We can interact further with Professor Ralph as we go out on for break. We will take a short break by taking a group photo with our Vice Chancellor and our keynote speakers and all the delegates, and will be directed by the Secretariat outside. But before we do that, we will take a few instructions from James. So since we have new guests and delegates who arrived when we were already proceeding, I want to invite them to mention their name. And we'll start with our keynote invited guest. Give us your name and your area.
Good afternoon. Sorry for coming late. My name is Rispa Torore. I'm a senior lecturer in the Department of Immunology. Actually, Immunology Section, Department of Pathology, School of Medicine, here at Moi University. And my area is genomics of cancer and non-communicable diseases. Thank you very much for inviting me.
Karibu sana, Dr. Rono.
My name is Dr. Selina Rono from University of Eldoret, Department of Biological Sciences, medical microbiologist. Thank you.
Thank you. I think we also have Ezra.
Good afternoon. I'm Kirare Ezra, early career researchist, aiming to specialize in molecular plant pathology. Thank you.
I think that's all for now. So when we'll take our photo from outside here, we also have bags. When you're walking out, pick your bag from outside the reception here, and we will be directed from outside on where to go for our group photo. Thank you very much, everyone, for being here. Karibuni sana. On the same. So we have Feng online, ready to present. Feng, hello Feng. Hi. This is Emily, and we have all of us, the delegates here. We are ready to listen from you. So we'll give you 20 minutes to do the presentation. Yes. Yes, it's visible.
So my work is mainly work on the African electric fish. I'm more interested in the genomics of their communication signal divergence. So I think you heard a lot about electric fish this morning. So you heard about electric eel and also electric ray. Actually, in the world, the electric fish has been independently involved at least 6 times. So there are 6 independent lineages in the world of a different type of electric fish. And those electric fish, they have very specific electric organs, which mostly derived from their skeletal muscle. And they use this a specific organ to produce the electric organ discharge. We call it EOD. And EOD can be used for navigation and communication, and some extremely cases for predation. If you look at the histology of electric organ, you will see that it's composed by a lot of well-organized cells. We call it electrocytes. And each electrocyte can generate their micro EOD due to the ion currents across the anterior and posterior phase membranes. And the sum of all micro EOD from all electrocytes then is our measurable EOD that we know. We are interested in one of the 6 independent lineages, Momyroidae. And if you are familiar with the evolution of teleosteofish, you might know that this family, it belongs to the superorder Osteoglossomorpha. And in Osteoglossomorpha, it is usually very species poor. But only in this family, Momyroidae, there are around 200 species that are described. And such adaptive radiation in Momyroidae, people think, it might come because of their very divergent EOD. And for example, in one of the genus Campylomyrus that we are interested, there are around 16 species that sympatrically distributed in Congo River. And each species, they have their species-specific EOD that varies in both duration and also their shape. So you will find some species that have biphasic EOD, some have triphasic, and even fourphasic EODs. And also the duration that varies. It can be like 100 times. Some species like compressed rostrus, they're very short. It's only some 100 microseconds. But in some species, like the Rhynchophorus, it can be over 40 milliseconds. And one of the function of EOD that we found from our group that it can affect the mate choice. This is a behavior experiment that's performed by our prior PhD student, Rebecca. She used a focal species and put 2 types of EOD on the side. One is conspecific EOD species and one is a heterospecific EOD species. And she found that during the breeding condition, the males always shows a very significant preference to the conspecific EOD species, suggesting this might be a male-driven mate recognition. So that suggests that the EOD could be possible that be a prezygotic isolation mechanism if it really can affect the mate choice. And I used 41 wild catch samples for whole genome resequencing and tried to build the phylogenomics of this genus. And what I found is almost all the species, they have monophyletic topology. Each clade, they have only one species and a very stable EOD in each clade, except for the one big group Alces. But inside Alces, there are 3 subclades, but each subclade, they also have very specific, very unique EOD as well. And apart from the species that we already know, there are also 2 other new species that is potentially exist and with a very distinct EOD species, EOD type as well. This suggests that the adaptive radiation that can be still going on in this in Campylomyrus and the EOD, that it might contribute to this rapid radiation. And I tried to detect the gene flow among this genus using the whole genome data, and I only found a very weak gene flow that from species Numenius to another species Curvirostris. So if the EOD actually is very distinct among species and very few gene flows among those sympatrically distributed genus species, it is very possible that the EOD can really play a prezygotic isolation in speciation. And if the EOD is such important in speciation, that's why the EOD has such big divergence. That's what I was interested as well. So I firstly had a look at the divergence at the EOD duration. You may recall that the EOD among the Campylomyrmex that can vary 100 times. There are some species like compressed rosters is only 400 microsecond, but in some species, Rhynchophores, it can be over 40 milliseconds. So I performed RNA-seq and compared the gene expression of the electro organ and also their skeletal muscle. This is where the electro organ derived from in different EOD duration species and also their cross-species hybrids. From the principal component analysis of the RNA-seq data, I found that the PC1 can really separate two tissue type. That makes sense. But the PC2 that shows a very clear pattern that relative to the EOD duration. So we have short EOD, medium EOD, and the long EOD. This suggests that there might have a bunch of genes that shows with the expression that relative to EOD duration. That's what I did next. I tried to cluster the gene groups with different expression that shows significant relationship with the EOD duration. And eventually I found 3 gene groups that with different either increasing or decreasing gene expression across the EOD duration. And there are some candidate genes that I found for the EOD duration, mostly these potassium channel genes and also some transcription factor. Very important is this gene that Ralph mentioned about in the morning, is this KCNJ2. It is highly expressed in all the electro organs. Also, the expression is higher in elongated EOD comparing to short EOD. So this gene was not only found in the purebred species, but also in the hybrids. Maybe a little bit background to the hybrids is we did a lot of artificial breeding to cross the different species. And very interesting is whenever we cross the species with short EOD, with elongated EOD species, the F1 hybrid always show the EOD always shows a preference resemble to the elongated EOD species. So this is very interesting. We thought about why this happens. One of the hypotheses we thought is since the F1 hybrid, it contains 2 subgenome from the parent species, it is very likely that the alleles from in F1 hybrid from the alleles from the elongated EOD species, it shows more dominant expression comparing to short EOD allele. That's why we perform this allele-specific expression on these hybrids. But actually, the results is very opposite to what we thought. All of the genes that showed allelic expression imbalance that shows a preference to the short EOD alleles. And there's only one exception, is this KCNJ2 that we found before in the last slides. That it is the only gene that showed the dominant expression on the elongated EOD alleles, suggesting that this gene can be a very strong candidate that effect involved in the EOD duration. And that's the EOD in the adults. This gene was also found in the EOD autogeny as well. You may recall the introduction from Ralph this morning that most of the species in Campylomyrmex, they started with the same juvenile EOD, and the short EOD species, compressed rosters, it stays all the same from to adult EOD. But some species like Rhynchophores and the F1 hybrid, the EOD duration will be elongated and then reach the adult EOD. And what we found is this KCNJ2, the expression of these genes, is highly expressed in adult EOD comparing to juvenile EOD in Rhynchophores and also F1 hybrid. And very interesting is in F1 hybrid, the expression of this gene, the allele from the elongated EOD species, is getting more and more dominant during the development, suggesting that the expression of this gene might also affect the EOD development as well. And because this is the most powerful candidates, we amplify these genes in different electric fish and also non-electric fish. What we found is there's a disruptive selection that happens in two electric fish clays. And very interesting is these two clays, both of them, they have very elongated EOD comparing to the others. And there's one amino acid happened at the selection size that it caused amino acid change from D to N. This was also happened in human disease as well, that caused a mutation on these transmembranes in this inward potassium channel. So we thought about if this is a disruptive selection, and this selection can really affect the EOD duration change that caused them, it may favor the either short EOD or very elongated EOD, but not intermediate EODs. Yeah, this is about the duration divergence. I'm also very interested in the EOD divergence in the waveform, the shape as well. That's why I resequenced some samples from the compressed rosters. In this species, it has two types of EOD. One, they have the same length, duration, but there are two types of EODs. One is biphasic and one is four-phasic EODs. And from the genetic data I resequenced the samples from compressed rosters with the two types of EODs. And what I found is from the principal component analysis, those two types of EOD cannot be separated genetically. So it is possible that there are not many genes affect the EOD waveform at the same time, but possibly only few genes that really affects them. So that's why I try to quantify the genetic differentiation among those two groups by calculating their FST. Firstly, I use the sliding window to locate the candidate area in the genome to find the possible context in the genome. And then in the high FST context, I try to find the very high FST SNPs that we target several candidate genes. There are two very possible. One is this JAK1B gene and also TNF3. Both of them, they can regulate the cell differentiation and tissue pattern during the development. This is still very new data, very preliminary data. So we're still thinking about what to do next to check about these genes in different species in Campylobacter, and also have looked at the expression of those two genes in different EOD species. So coming to the end, so what I did, we tried to use to have a look if this EOD very contributes to the prezygotic isolation. So we found actually the taxonomy is still under debate because we still find like new species, and all of the species, they have very divergent EOD and they are monophyletic in phylogeny. So it is possible that this adaptive radiation, they are still going on, and the EOD might really contribute to the rapid radiation. The second part is about the EOD divergence among the species. So we target several candidate genes, like especially the candidate genes for duration, like in the case NJ2. Also the candidate genes for the EOD waveform, like JAK1B genes. So still, as Ralph mentioned in this morning, we are still doing some functional tests for those genes to try to knock out those genes using CRISPR-Cas and to see if they have any really function change or not for those genes. Yeah, so coming to the end, thank you very much for the organizers first, thanks for James, and also thanks for my supervisors, Professor Ralph Tiedemann and Professor Frank Cashbaum, and also some collaborators from University of Potsdam, Humboldt, and Namur. Thank you so much. Yeah. NJ. Yeah, there was some founding that the genes can affect not only on the signals but also in the facial the cranial it's called facial structure. That's what also we are interested about because in our Campylobacter there's a very big difference in the SNOD as well. We were wondering if these genes will also can be affect the function on the SNOD change as well, because some samples, some species, they found this KCNJ2 that they have they might have a function on the cranial this facial structure. That's also what we are interested as well.
Where you show us the allele-specific expression, which, when I remember correctly, this is based always on intraspecific crosses, interspecific crosses, right? So do you perform the crosses in both directions? So the female of the one species and the male of the other, and then the other way around? So can that also be, in the worst case, be some like sex-specific expression?
From our the results that we had now is only from one direction. So unfortunately, we don't have the cross hybridization from another direction. That's what we are also interested about. But we're not sure if this because of the sex, because in our species in Campylobacter, all the species. In all the species, the EOD, they are consistent in both sexes. So we are not, I don't think, we are not sure if this could be a sex-linked things or not. And this is what we are trying to do, because we try to find some sex-linked genes, but there's, at least we didn't find any sex-linked chromosome. But possibly there's some sex-linked genes, and this is what we're going to do, and if it is linked with some candidate genes that we know or not. Thanks.
I was just wondering if you've considered or have already done it, comparing the genome of non-electric fish to this, or any non-electric fish that is a common ancestor that you can compare and then see if there is anything we can learn from, or you can learn from the genome evolution perspective. And another question, which is maybe a naive question, is related to your RNA-seq data. I don't know at which stage you can detect the EOD or the electric discharge, but I was just wondering if you have considered collecting sample at different growth stages and then compare the RNA-seq and see what is going on in terms of gene expression, and if these KCNJ2 gene can be found at different stages or something like that. Thank you.
Yeah, thanks a lot. That's a very good suggestion. Because raising the fish really takes some time. As from juvenile to adults, it can take like more than 2 years. So for now, we only had one stage for the transcriptomic data, and we are actually not sure if or we thought about to sample the more the samples from different life stages, so to see there's more detailed or information about the gene expression. Yeah, but I think this is one of the plans for probably in next stage. But for now, we haven't we are still raising the fish, so yeah, it will take quite some time, but it's a good plan. And regarding your first question, I'm really sorry I didn't hear it properly. So if you may repeat it again, or
about comparing the genome of these electric fish to an electric efficiency, if you can learn anything related to genome evolution or yeah, something. Thank you.
Yeah, thanks. And I think that there was some analysis before from Professor Jason Gallen from US. He did some comparison of different the genomes from different electric fish and to see there's any convergent evolution. And he did found some interesting results, like for example, there's both potassium sodium channel genes like KCN7 and also SCN4A, and those genes, they are very much very important in almost all the lineages, electric fish lineages. But still from the recent whole genome data is still very limited, especially in Osteoglossomorpha. So now I'm right now sequencing another genome from Myroidy. Hopefully, it can help to solve other have more interesting results. Thanks. Thanks.
For the nice presentation.
Thanks, Emily.
Yes. We want to welcome our next presentation, and our next presentation is Sarah Waswa. Please, you can step to the front and begin your presentation. You'll do a 15-minute presentation, then we have 5 minutes for interacting with you. Thank you. Those who are online, if you have a question, you can always raise up your hand, and the technical staff will just notify us to give you an opportunity. Sarah, welcome to the presentation. Sarah, you will introduce yourself, and then you take over. So for those who are next presenters, you can load your presentation slides. Talk to the IT team so that it is downloaded on time for presentation. The IT team, please make sure that you download all of them ready for presentation.
Good afternoon, ladies and gentlemen. My name is Sarah Waswa Nahumicha, an MSc student at the University of Eldoret, and I'm currently based at the Walter Reed Army Institute of Research in Kisumu. Today, I'm privileged to present my MSc project, and it was on virome of bats in the Mount Elgon region. Today, we are going to learn or see how genomics is applied in relation to public health. So, to start off, bats are the largest reservoirs of zoonotic viruses, that is second after rodents, and some of these viruses, they include the famous Ebola. We have SARS-CoV, that is as we have seen in recent past, the coronaviruses. There's the Nipah and Hendra viruses. And the bats, they cover approximately 25% of mammalian biomass. That is, they are the second after rodents again. And why we chose Mount Elgon region, it is known to host extensive cave systems that harbor dense bat colonies. And this region is on the Kenyan-Uganda border. And Mount Elgon region, historically, it has been linked to the Marburg virus spillover events, that is in 1980 and in 1987. And currently, there is some work that is being done around the region to see if we can detect Marburg virus, if we can still detect it as of now. And so, the full viral diversity of these bats in the region, they remain largely uncharacterized. And so most of the surveillance, most studies actually have focused on detecting a non-virus instead of uncovering the full diversity of the viruses in the region. And so we opted to do this study to understand and improve our knowledge on the complete virome that is mostly critical for predicting and preventing the next zoonotic spillover event in case it happens, which we don't want it to happen. And so the objective of the study was to characterize the full assemblage of the viruses in these cave-dwelling bats using untargeted metagenomic sequencing. So our methodology included 3 processes. That was collection of the samples, processing, and then analysis. And so on field sampling, mist nets and hand nets were deployed onto the entryways of the caves, and we were able to capture 86 bats from 3 caves, and the samples that were collected were the oral and rectal swabs. So from all these 87 bats, we were able to get samples, oral and rectal swabs from all the bats. And so capture and release strategy was employed. After collecting the samples, the bats were released back to the wild. We made sure not to harm the bats, in case anyone is wondering. Ethical approval was sought prior to fieldwork. And after the samples were collected, they were transported to a laboratory in Kisumu. And subsequent preparation and subsequent processing of the samples was done. That is, initially the samples were extracted, the total nucleic acids were extracted, and library preparation was done using the Colibri kit. And to identify the full virome diversity in the samples, we opted to enrich our samples with the probes. They're called the VIRCAP-SEC VAT probes. Those are designed by the University of Columbia. They're able to detect over 2,000 viruses. So that panel was used to enrich our libraries to get a good coverage of the viruses. And then sequencing was done on NextSec 2000, Illumina, and it was untargeted, that is unbiased. We didn't know prior to it what we were going to get. And analysis of our work, the RIM pipeline, that is the rapid identification of microbe pipeline, it is also designed by the guys at the University of Columbia, was used to classify the viruses. To identify the host species, a custom bioinformatic pipeline was used. And so on to results, we were able to identify 6 bat species. And it is important to note that out of the 87 bat species that were captured, only 77 bats were able to be classified. And it might be because of the pipeline that was used to do the analysis, and that was, we used the barcode database to identify the bat species. So it might be because the mitochondrial DNA that was mined didn't have the COI region in it. That's why maybe some of the bat species were not able to be identified, or some of the assembly genome were not able to be classified. So the species that were able to be identified, that was the Cholera afra. It was the most dominant species in the region, and it was detected across all the 3 cave sites. We also had detections of the Miniopterus inflatus, that is the greater long-fingered bat. It was also identified mostly in 2 caves. We had detection of the Nysteris thebaika, that is an Egyptian slit-faced bat. And we also were able to classify the Hipposideros ruba. It is mostly identified by its round leaf nose. We also were able to identify the Rhinolophus eloquens. It is distinct by its distinct horseshoe nose. And we're also able to identify Lisonysteris angolensis. It is an Angolan fruit bat. And out of these 6 bat species, 5 of them, that is the Cholera afra, Miniopterus inflatus, the Nysteris thebaika, Hipposideros ruba, Rhinolophus eloquens, all of them are insectivorous bat species. Lisonysteris angolensis is a frugivorous bat species. It's to mean that all the 5 species, they feed on insects mostly. Those are the moths mostly in the caves, and other small insects. And Lisonysteris, it's a frugivorous bat species, meaning that it mostly feeds on plants or fruits in the area. And after detection of the bat species, this was done with identification of the COI region in it, in the mitochondrial genome. So this identification, as I'd earlier mentioned, it was through a custom bioinformatic pipeline. And the COI region in it were able to identify those. And this phylogenetic analysis, it was also based on the cytochrome oxidase subunit 1 gene. And in red font, it might not be that much visible, but in red font are our study sequences. In black font is the NCBI sequences. So we were trying to see if those results from bold can complement NCBI findings. And it was seen to complement the results. Most of these study sequences in red, they belong to the Cholera afra. There are 6 clusters that are complementing our previous results. On to viral family prevalence. Out of 87 bat species that were sequenced, 71 had hits for viruses, and most of the viruses that were identified belonged to the family Retroviridae. 77% of the bats had Retroviridae. We had detections of the Parvoviridae. We had detection of the Genomviridae. We had detections of Herpes viridae and Paramyxoviridae. In low abundance or in prevalence or occurrence, Rheoviridae had the lowest detection in all the 71 bats. In total, 15 viral families were detected, and the Retroviridae made the most of the virome. So when you break it down, you can see we were able to classify 20 viral species, and these are just only top 8 of the viral species that had high abundance or high prevalence, sorry. So at number 1, it was bat gamma retrovirus. It had an percent occurrence of 73.24. We had also densovirus. It is an insect-associated virus. We had gamikibivirus. There was also detection of the bat astroviruses and bat coronaviruses, though it was the alpha bat coronavirus. And nodaviruses at a 5.63% occurrence. This is a stacked bar plot that is showing the relative abundance of the viral species across the individual sampled population. And so in purple color, it is the bat gamma retrovirus. It was the most dominant, as I mentioned earlier. And these are the endogenous, mostly it is an endogenous viral element, meaning that they coexist with the bats. And they're often unpathogenic. And they mostly serve as an evolutionary record of the ancient viral exposures in the bats. There's also Parvoviridae. Those include the Densoviridae, and as I'd mentioned earlier, it is an insect-associated virus, meaning that it might be due to the diet of the bats in the region. So that's why we were able to see them. And then Genomaviridae, it is mostly a transient virus. So it is important to note that we also had detection of the rotaviruses. We had rotavirus C, D, and H. And those are known to cause the gastroenteritis mostly. And a highlight from this bar plot is showing that the heterogeneity of this viral composition shows the multivirus presence within the Montelgon bat population. As it can be visible here, you see that some of the bats have more than one virus in it. That is, it's showing the co-infection of the viruses in the bats. This was to see the relationship between the host species and the viral species abundance, which species has the most viruses in it, or which one has the most abundance of the viruses. And even though Cholera afra was the most dominant species, it is not harboring a lot of viruses as compared to Miniopterus inflatus. On this bar plot, you can see that Miniopterus inflatus has more viruses. Those are the beta herpes virus, trace delta papilloma virus, and the bat coronavirus. And it is distinguishable from the remaining species of the bats. Neisseris thebaika is standing out with the rotavirus C. I think I chose the wrong colors. But you can see that across all the bat species, retroviruses remain the most dominant viruses in them. What can we learn from these results? There is minimal zoonotic implication of these viruses. It means that even though they are at a low abundance, it is not negating the fact that there is potential for them to cause a zoonotic as spillover event. We just need to be more vigilant with it so that we know what we are dealing with. So we've seen that we have the Paramyxoviridae, they have the zoonotic implication with it, and the Coronaviridae, we had the SARS-related lineages, although we need to do more complementary testing to be sure that those are the coronaviruses that are able to cause a major pandemic if it comes to that. There is also cross-species transmission potential and high co-infection rates. Those are multiple viruses within a host, increase the recombination risk, and it can lead to novel viruses that have zoonotic implication. And so, in light of these results, what we can advise or recommend further is to do a longitudinal sampling across the wet and dry seasons, or to increase our sampling strategy so that we can cover a lot of caves, a lot of bats. We can see their patterns across different seasons. We also recommend full genome sequencing of the novel viral species. And we also recommend collaborations across different multisectoral departments to do a cross-border surveillance network, because this is critically important in relation to public health, because we don't know what the future holds, but as scientists we can try and figure that out before we get to that point of an epidemic or a pandemic. And we also need to do community education or to increase awareness on the neighboring people, because these caves are situated where people are, like multiple households, they live within that region where the caves are. So it is upon us as people who do this research to improve or to create awareness to those people that regardless of these parts, they can be harmful or they can be beneficial to us. So we just need to do better as scientists. Thank you.
Thank you. Thank you for that presentation. Let's clap for her. Yes. Thank you for that presentation. I know we had said next presenter is Ezra, but we have Hills, who is online, and we can welcome his presentation. Was supposed to be just before lunch, but now let's give him an opportunity. Sarah, thank you so much. I will allow you to sit, or then we can ask questions at the same time for everyone, for those who are physical. Yeah. Thank you. Clap for Sarah as she sits. Yeah. Our next presenter is Hills and is online. So let's allow that presentation to run. Hills, are you there? Okay. Welcome. We give you 15 minutes. Yes, we can see your presentation. Please proceed.
All right. Okay, thank you very much. I'm Hills Kabuola. I'm a student at my research it focuses on the genomic morphological characterization of loudspeakers, locally here known as growth. Are you able to hear me?
Yes, it's clear. Please proceed.
Okay, very much. So this is part of my master's thesis. So it focuses on the cryptic genomic, recipe, and ecological functioning of loudspeaker catfish. So background, says belongs to the country. It has approximately over 200 species across the sub-Saharan Africa. These are nocturnal and herbaceous with characteristics stimulation. On this part is like they produce sound using their fins. So that's why they are called speakers. Lake Malawi is globally celebrated for its secret radiation. It has over 800 species which have evolved within the lake, representing one of the most dramatic examples of adaptive radiation known to science. It attracts scientists all over the world. But however, the lake's non-secret fauna remain largely understudied. Cynodontis injasi is one of the most ecologically important fish species in Malawi. As we can see here on the graph, during the Biomass 2023 survey by the Department of Fisheries, it was in the top 10 of the species which were caught during the Biomass. But it's largely given less attention. In 2006, Cobb Muller analyzed the 900 mitochondrial fragment of the Cynodontis species. The indicated monophyly of the East African Cynodontis clade is nested within Central and West Africa representatives. Based on phylogeography and phylogeny, and they have a hypothesized that synodontic was colonized independently, unlike the other haplochromized cichlids, which were colonized from in Lake Tanganyika. But the previous molecular work was limited with small fragments and small sample sizes insufficient to resolve the population level structure. So, and Ganese et al. In 1990 documented intraspecific karyotype variation in the genus, which evidence that molecular diversity may exceed what morphology suggests. So this is common across this, African freshwater fishes. Just to rely on morphology alone routinely fails to resolve evolutionary history. So what we needed is advanced genomic tools to detect reproductive isolation. So our hypothesis of these species were the spot patterns. So visible variation, the local fishes and so the taxonomists noted varying spot patterns in Sandonotis in Jasai from entirely spotless to spotted. Our initial question was, do these different spot fossils act as diagnostic markers for distinct characteristic genetic regions? The challenge was to rely on the body shape of spot pattern alone, as we have seen that it often fails to resolve evolutionary history in African freshwater fishes. So our solution was to get advanced genomic tools where advanced genomic tools were required to detect reproductive isolation and cryptic diversification, which underlying this catfish. So our questions and objectives. Our research question was, is Cyanotis just one species or a complex of multiple cryptic fishes? So our objectives was to first generate common level reference genome as a foundational resource, as we could not rely on fragmented genomic data. Fragmented assemblies often miss structure very critical for understanding reproductive isolation. And also, our second objective was to characterize population genetic structure using whole genome sequencing. Resequencing of the collected individuals to identify cryptic lineages across the lake. So we sampled individuals from different depths and location to assess population structure. Our third objective, so that we needed to map this the genetic data back into the physical world, like do the genetic differences manifest in body shapes, spot patterns, or depth preferences. So we were guided by those three objectives. So we did electric sampling, Malawi, we sampled in Karonga, we sampled in Kata Bay, Kota Kota, Salima, and also Mangoji. Our sample collection, we had two sampling projects. First, we were collecting fish from fishers, which were this was near shore sampling. We're collecting from Zano fishers, and they were telling us where exactly they casted their nets. So we were able to trace where they caught the fish, and we were able to, guided by them, we were able to go and where they went to get. The second sampling was we joined the Department of Fisheries in the biomass throw service. We did so we were able to get samples all over the lake from the deep water ocean. Our sampling procedure so each specimen was photographed under the standardized lighting conditions in a photo tent to record external features, including body specimen in 0.1% S222 solution in accordance with the proven ethical procedures. So we for the genome sequencing, a high molecular weight genome was extracted from the fresh frozen Sandonotis jasai and sequenced on the Oxford Nanopore technology platform in Cambridge. The library was sequenced on promethion flow cells, which yielded about 72.2 gigabit data, equivalent to approximately 79 times genome coverage, which read about it was a contagious genome. So then also we integrated morphometrics in the research. We did morphology for the right and page system of measurement. So successfully generated the first chromosome of the entire family of Machokidae. The assembly contagious chromosome ranging in length from 58.1 megabase pairs to 22.7 megabase pairs. The mitochondrial genome was fully recovered, quantic of 16.5, 16,529 pairs. Only 6 minor scaffolds remain, representing less than 0.2% of the total. Were then mapped back to this reference for consistent variant calling. So here we have the percentage of variance explained for the first 20 principal components. Yeah, here. So PC1 captures the largest Oh, okay. Can you hear me now? Are you able to hear me now? All right. Oh, as you can see on the second plot, the PC3 and PC1 further separates C from D. So I plotted a tree, nuclear phylogeny, which cleanly resolved 4 bus grades, consistent topology and genome-wide. Then after doing that, I tried to plot a mitochondrial genome. So we know mitochondrial genome is materially inherited and does not recombine. Here, the nuclear and mitochondrial phylogenes do not agree. This is not an error. It is a biological signature of complex evolutionary history. So there should be 2 processes driving this quadrant in here. It could be incomplete linear sorting of ancestral polymorphism, sorted incompletely, or in progressive hybridization. I did plot a sliding window PCA for the chromosomes. This is chromosome 2, of course, out of the 27. This is chromosome 2, which showed consistent separation of the clades. And here, interestingly, here you can see this structure here, an inversion in chromosome 2, which is consistent in clade B only. Morphological trace plot, the corrected size is dependent variation of ratios. This analysis reinforces the pattern of extensive phenotypic overlap while revealing clear signal of divergence in the lineages. Here's just a sample of the clades, clade A, B, C, and D, how the morphological pattern. Ecological distribution among clades, clades A and B were found everywhere. You can see here and here that through the 2 processes of sampling, clades A and B were found everywhere. But clades C and D they behave like side-attached Nguna cichlids, completely absent from all deep water troll catches. And we see here, I put the PCA by location. So there are these two species in deep and shallow areas, and also, but see and restricted, one to the northern part of the lake and the other one to the southern part of the lake only. So the challenges, the conservation implication, we know the current status of the Cynodontis njasa is least concern, but when grade C and D are isolated to specific shallow littorals, or making them highly susceptible to nearshore segmentation, a gradual runoff, and even illegal beach seining, we could lose these unique evolutionary lineages representing thousands of years of independent evolution. So those are the, the conservation implication of the Cynodontis catfish. For policy and management recommendations, our findings are not just academic, they have immediate implication on how Malawi manages its fisheries. The evolutionary significant units, they should be recognized pending formal taxonomic revision. These four lineages must be treated as distinct evolutionary significant units. Lineage level management, there's to shift from species level to lineage level management to prevent the silent extinction of cryptic specialists. And also spatial protection is needed to management and protected zones around vital littoral rocky habitats, such as in the northern part of Lake Malawi, where there's no Lake Malawi National Park there. So it should be focused. Also, future genomics should expand genomic audits to other economically significant morphological static factors. For example, the Bagras meridionalis is also mentioned as only one species in the lake, and also prioritize the functional characteristics of the chromosome invasion to understand its role in adaptation to deep water pressure or reproductive isolation. So there's a clear and exciting roadmap for future research that extends naturally from our findings, characterize the gene content and functional significance of the chromosome 2 invasion. Candidate processes may include the depth or pressure adaptation or osmoregression and reproductive isolation maybe. Incorporate an outgroup taxon, definitely root the phylogeny and establish whether the generalist or the specialist trait is ancestral, or did the shallow water ancestor conquer the deep, or did the pelagic fish adapt to the rocks? So, and also undertake a formal taxonomic description of the four lineages as distinct species, including examination of historical type material, extended meristics, because we didn't do meristics in this study. So it would be another future outlook for this research. On that, I say thank you very much for your attention.
Thank you so much. We'll take a few questions. We'll start with his presentation, then we ask questions and comments for Sarah as well. So all of us, do we have anyone with a question or comment for this presentation? Yes. We have experts in cichlids, hills, we are here, and we have a few questions for you.
Yeah. Okay,
Yeah.
thank you.
Yeah, so thank you, Hills. So from your clustering, how many very clear clusters did you identify, and what is the level of exploitation of this catfish in Lake Malawi? Thank you.
You respond?
Okay, yeah. From the clustering, here we had like 4 clear clusters of the species. That's why I plotted PC1 versus PC2, whereby I found that the two other clusters which were very close, we thought maybe this would be very close. Then I also plotted PC1 versus PC3 to see if there is that difference. So you can see from my first graph here, this grade here, C and D, they were very close. So I did plot here PC3 versus PC1 to show if they were that different. So here we can see clear that they were that different. Yeah, so it yielded about 4 clear clusters. And to answer your question about the level of exploitation of this species, currently this species is found even in the markets. Previously, it wasn't being found in the markets, but now these days we can find them everywhere. Every market, every fish market is there. So we can say the level of exploitation is very high currently compared to previous, compared to the previous years.
Or comment?
Okay.
Anyone with a question or comment? None. Thank you so much. We clap for him again. Let's clap for him. Interesting. As we usher in the next presenter, who is Ezra, we can take 1 or 2 questions for Sarah. Sarah's presentation earlier was on bats and genomics of bats. So as we set up Ezra. Yeah.
Thank you. Very nice presentation and also very important topic. I just have one question, and I wanted to ask you, you collected oral and fecal swabs. Did you compare those 2 swabs, like with the findings you had in those swabs?
Thank you for your question. When we were collecting the samples, we had 3 sample types. We had blood, we had fecal pellets, we had oral and rectal swabs. The oral and rectal swabs were being pulled into one vial. So when we were processing the samples, we were doing it as one sample. Yeah, we didn't compare between the different sample types.
Thank you. Any other question? Yeah, we come here, then we go to.
Thank you very much, Sarah. I'm just curious. So bats are finding themselves in our homes. Here they are known to be living in the wild. What could be driving this? Or did you attempt to find out why they're coming to live with humans? And what are the implications? Thank you.
Yeah, they're finding themselves in our households because one of the reasons is we are encroaching into their habitats. So when we are cutting down the trees, some of the bats actually live on trees, some of the bats live in our rooftop. So when we go into their habitats, they're forced to come to where we are. Yeah, and some bats, even in Maseno, when you go to Maseno, some bats are in our houses. Implication on One Health. I would say we have developed, or rather they have developed the system, our immune systems. There's some study that needs to be done on why we can coexist with those bats in our households. Because in the long run, it might lead to serious public health concerns that can cause serious adverse effects.
Thank you. Who else has a comment or question?
Yeah, thank you, Sarah, for that good presentation. In your presentation, you raised the issue about was it abundance or prevalence of the viruses you obtain from the bats. And the highest was the viruses of the family Retroviridae. And I don't know whether this is of any concern, because we are aware, for example, the family Retroviridae, as far as I'm concerned, has 2 viruses of medical importance. One is HIV, and the other is human T-cell lymphotropic virus that causes leukemia. Now, the fact that there was high abundance of viruses of the family Retroviridae, of what concern or of what significance did you attach to this? And do you find that this is a threat to humans who are living in that place?
Thank you, Dr. Salina, for that question. The viruses belonging to the family Retroviridae that we were able to identify specifically were of the bat gamma retroviruses and the bat beta retroviruses. So as part of the recommendation by my supervisors and the lab, we need to do more complementary testing to see if those viruses that we identified in our samples have potential for zoonosis. I wouldn't say for sure that we are not going to negate the fact that they have the high possibility of infecting mammals when we are infected with them, but we just need to do more research on it.
Yes, thank you for that interesting presentation. Let's clap for her. She did very well. As we usher in Ezra to do the presentation, 15 minutes from now.
Okay. Thank you for an opportunity to make a presentation. And we are looking at the diversity of genetic diversity of Pseudomonas axonobodies, Patopha ficnicola. This is a study that was done between the year 2022 and 2025. And you take note that Pseudomonas is a bacteria genus that occurs worldwide, and it causes diseases to a wide range of crops, including the legumes. Generally, all the cereals, they are mainly infected by Pseudomonas, whereby they cause the various diseases, resulting in the reduction of both the quality and the quantity of the harvested parts of the crop. The Pseudomonas has got more than 20 genomic species that have been identified so far, and they show some great degree of decrease of genetic general sequence resemblance as identified, and also the members of Pseudomonas exhibit host specificity and a wide range of phytopathogenic potential towards other crop members of different species. And also, Pseudomonas in Kenya has been reported to infect several crops. Of interest in our research was the cowbees, which has been associated with which the bacteria has been associated to cause bacterial plague, and it has led to reduction in both the quantity and the quality of the harvested parts of the crop. In Kenya, cowbee is mainly utilized as a vegetable in most households, or it's used as a fodder for the livestock, or also the grains are consumed in other communities as legumes. So when the quality of the harvested parts are affected, even the communities depending on the same crop for economic benefits will suffer, as the returns will be reduced. Pseudomonas has been associated with genetic variability that has resulted in a challenge in management of the disease. And here in Kenya, even the disease is still reported as the main cause of reduced quality of both quality and the quantity of harvested parts of the cowbee. So in light of this, our research was mainly to assess the genetic diversity, of the Pseudomonas axonobodies, Pataphaphic nicola, using in the simple sequence repeat markers. And in this case, this is a pure culture of Pseudomonas axonobodies, Pataphaphic nicola. This was from the lab, from our lab, the molecular fish genomics lab. We isolated the pathogen until we got the pure cultures. All these are Pseudomonas axonobodies, Pataphaphic nicola isolates. The A shows the typical yellow color, and it's as a result of sandum produced by Pseudomonas, while P is the creamy yellow one. It's still an isolate of Pseudomonas axonobodies. C shows it's a light yellow in color, while A and B are the typical yellow ones, while C it's creamy yellow, and for D it's light yellow in its pigmentation. These are pure colonies of Pseudomonas axonobodies as isolated from fish genomics lab. And then this is just a summary of the protocol that we used in isolating the DNA in the lab. And we remember we used a DNA extraction kit, and in this case it was a Simoclean one that we used. Those are the conditions that we followed. And then what is shown here, this is a pure DNA. Once we isolated the DNA, we ran through the gel. We wanted to check for real if the DNA was present after we isolated. This is just a pure DNA without any amplification. It's just a pure DNA of just, we sampled not all the bacteria that we were interested. We just sampled a few to just represent the rest. And then when we did amplification, the amplicons, these are the amplicons as you observed in the gel documentation. These are the different isolates, and these are the identification as we had coded. After running the amplification, we send our samples, amplicons, to Incapa Biotech for sequencing, and all the isolate showed identity to previously identified Pseudomonas axonobodies, Pataphaphic nicola, that are present in the NCBI gene bank. And then This is the procedure we followed in doing analysis of genetic diversity. And then we followed the initial method we indicated earlier for DNA isolation. And then these are the markers. These are the markers that we used. Initially, we tested a total of 10 primers. And what was used, we only adopted those ones which showed polymorphism, while those ones which never showed any polymorphism were dropped. So we used a total of these primers. These were 6 primers that we used. We had tested 10 of them, but since some of them failed to show any polymorphism, they were dropped, and these were the shown were adopted. Then we analyzed when we scored the bands. 1 was used to score for any band that was indicated, and then 0 for absence of a band. And then we analyzed the diversity using the NTSYS software. Then cluster analysis was done also using the same program. And then we did polymorphism information content using the pairwise population matrix of net genetic distance. And then we also did the principal coordinate analysis and molecular analysis of, and we've performed also analysis of molecular variants. This one is an image that we took from the gel documentation from one of the primers. The first image, this one, we had used D3 primer, while this other one, the second one, we had used it A31 primer, and all these were the same samples. They were the same samples. The difference here came in when we are analyze with the use of a different marker. But you can observe how the banding was observed. Despite they were the same samples. Now, what was the differentiating factor were the primers, and we can see the bandings here. So, similar to this, different primers, but the same samples. So we can see the polymorphism that was produced by the different primers on the same samples. So from this, we noted that McWane had the highest polymorphic loci, while the least polymorphic loci was observed in Meru. We mainly attributed this one to the number of sites that turned positive to the Sandomonas axonobodies. We sampled equal number of farms, but when we did analysis, there was a variance in the percentage of farms in each county that were positive. And this might have, the difference in polymorphic loci might have been brought in by the variation in the positive sites that were noted. These were the number of bands. The first one, this one, the blue one, represents the total number of bands that were observed in the different county samples. The first one is Isiolo, the second one is Kakamega, the third one is Laikipia, the fourth is Meru, the fifth is Makueni, the sixth is Uasin Gishu. So those were the number of bands. With Uasin Gishu, it lacked the private funds. There were no funds that were present only in Uasin Gishu. Then when we did analysis of molecular variants, we noted that the isolates, greater genetic diversity existed within the population rather than among the populations. And this might be attributed to manual spread of the pathogen through the movement of plant materials from one region to another. And then when we did principal or coordinate analysis, this is what we observed. And we noted that mostly, if we can see the spread of the populations, we can observe some that most of the populations from Uasin Gishu were mainly close to populations from Kakamega, indicating some close genetic similarity. Well, that as if, for example, we look at Makueni and Kakamega, the distance between the different points was large, indicating greater genetic diversity among the between the isolates. And then when we clustered, we noted that there existed two main groups, Group A and Group B, with Group A containing 47 isolates, while Group B contained only one isolate. Under under Group A, we had the different clusters. This is not a unique scenario to us alone, to our findings. The same has been reported by other researchers to have existed among the Pseudomonas axonobodys Butterfly fignicola isolates. As indicated, this dendrogram has two main groups, A and B, with A containing the different cluster groups, that's got 1 and 2, whereby most of the isolates are clustered into one group, indicating close genetic similarity among the isolates. While B is really isolated from the rest. And you don't know, is it related to, is it because of geographical? And if it's so, even the rest would have been separated like B. So this one, if we had funding, we would have done further analysis to check deeper. We would have done all genome analysis to compare the genome of this isolate that was in B in comparison to the rest. Then, in conclusion, we noted that there was a genetic variability among Pseudomonas axonobodys, Butterfly fignicola isolates, but the genetic diversity was not attributed to geographical location of the isolate, but rather to the differences in the individual population across the regions. That is the end of my presentation.
Yeah, thanks for the interesting talk. I have a question as to a potential biological reason for your final finding. I come more from the eukaryotic world, but is there something like effective population size? Is there a larger level of or a larger amount of bacteria where you find more genetic diversity, or is that far-fetched to think like that?
That question, any other question? Any other question? Let's take 2 questions.
Thank you very much for that nice presentation. Virginia Anculata is a very important crop, especially when you do the study. Drought-resistant crop, and very important for food security. I didn't see whether you did any relationship with your study and how much devastating is the disease on the crop. 2, maybe I want to know, in terms of diversity, how virulent the strains you got, how are they coping towards becoming more virulent, or whether maybe the crop itself is becoming more adaptable, and is there, where is it in the arms race? This diversity, so many strains you saw, which one is more virulent and which ones needs our focus if we really want to focus on ensuring food security among the people and all the household dependent on this crop, especially in the arid areas. Thank you.
Thank you, Ezra. Just a quick question. For diversity analysis of Xanthomonas, there is non-primers for analysis, that is REP, ERIC, and box primers. Did you consider that for analysis of diversity?
Okay, thank you for the questions. Let me start with the latest question, the last one. We never considered those primers. We never considered the said primers. And then the other one is when it comes to control. For the control, we do understand that the most effective and most promising control strategy is to approach it from the use of resistant crops. That is far much better because the pathogen, due to evolving genetic diversity and the changes that may happen within the genetic system of the bacteria, even if we develop control chemicals, some of these bacteria will develop mechanisms of either breaking down the active ingredients or evading developing strategies that they evade the control pesticides. The pesticides that we apply for control. And then for the extent to which it causes devastation to cow bees, since it's one of the key crops that are grown in Kenya, especially in arid and semi-arid areas. In our study, we never concentrated in estimating the levels of destruction caused by the pathogen to the crop. And then, on the question posed by Prof, when it comes to biological population, we only restricted ourselves to those sites which were positive. In this case, we had 48 isolates, and we only analyzed the 48. from the 6 counties that we sampled. But for more informative results, we can extend to the larger part of Kenya, especially we missed key counties that are growing the cow bees, like the Kilifi. We missed Kilifi, we missed Pungoma. This was mainly due to financial constraints. That's why we missed them. But if we had the finance, we could have even given a more elaborative results indicating the extent to which these pathogens are diverse. I hope I've answered your question. Thank you. Thank you for your time, and thank you for listening to my presentation.
Yes, thank you very much, Ezra, for that interesting presentation. We have our next presenter, Hilda. Hilda is next, and we request that the slides for Hilda are projected. Hilda will give you strictly 15 minutes. I've come to sit here so that when I show the 5 minutes remaining, you could take note. You have 15 minutes.
Okay, thank you. I'm going to do a presentation on the research was in improving taxonomic identification of enteromia species, that is Pisces cyprinidae of small water bodies of the North Rift region of Kenya through molecular techniques. Enteromias is a genus of small to medium-sized cyprinid species that are found in Africa. They belong to the family Cyprinidae, and we have approximately 350 species that belong to this genus, making it the most spacious and widely distributed genus on the African continent. So the challenges of studying evolutionary relationship in members of these species, that is enteromias, is their high degree of morphological similarity. They share morphological similarity, and this species plays a crucial role in local fisheries and aquatic biodiversity within the region. So the challenge in morphological similarity due to the shared physical traits, there's also convergent evolution. And these species sharing similar habitats, the species are often misidentified, leading to inaccurate data on ecological roles. Therefore, due to this misidentification of enteromia species, there is actually need for proper identification of these species, their distribution, and actually their adaptation to specific environments. These challenges, just as I've said, hinder formulation of effective conservation strategies, which should be species specific. The advancement of scientific knowledge leading to diversity, ecological significance within Africa, and also the challenge of overexploitation of the fisheries resources. Therefore, due to the need for species specific conservation strategies, molecular characterization actually helps to identify those species with low indices of genetic diversity so that urgent conservation measures are given. So the use of molecular methods, which we've talked about, are more powerful in identification of species and characterization, increases the chances of identifying cryptic species. These molecular methods actually generate more information that is important in management of these fisheries resources. Materials and methods. We did sampling, which we did selected randomly within the small water bodies of the North Rift region of Kenya. Sampling was done on man-made reservoirs and rivers that actually provide services to communities that live around these water bodies. Our aim was to cover a representative of all small water bodies in the North Rift, consisting of rivers and reservoirs that have an impact, that are impacted actually by agricultural activities. In reservoirs, samples were collected from 3 points, that is the extreme ends and the middle of the reservoirs, while in rivers, we did collection along the ridges. Selection of sites was based on accessibility and land use activities that are near to water bodies. So this is a map of the study areas showing the regions of the North Rift region of Kenya, that is the counties, Elgeyo Marakwet, Nandi, and Uasin Gishu counties. So the reservoirs and rivers that were sampled. This is what we were doing in the field, sampling using the electrofisher and also the say nets for the reservoirs. So upon collecting the fish, we did identification using the frozen pauli, and we were able to do fin clip. We collected the clips and preserved it in ethanol for further use for the DNA. In DNA extraction, extraction was done using the protocol, using the quick DNA mini prep as a protocol. After extracting the DNA, the pure DNA were preserved for PCR, which we did PCR amplification, and a total of 83 samples were sequenced for CO1, and the sequencing was conducted in Inkaba labs in South Africa. This is an electropherogram showing the PCRs of the species that were identified. After sequencing, we did data analysis and using Mega software, and these are the results. From the analysis, we were able to get the results from the basic, from BLAST, and we were able to identify 4 species, that is Enteromias numeri, Enteromas sarcops, Enteromas aplorogramma, Enteromas paludinosas. So from that, the percentage identity actually went up to 99.1%, showing that the CO1 actually gave us the 4 species, and due to the high percentage, it shows that it's a good method for identification of the fish species. So those were the 4 species that we were able to identify within the small water bodies in the North Rift, that is sarcops, aplorogramma, paludinosas, and further identification was done using the maximum likelihood tree, which clustered the species into 4, Numeri, sarcops, paludinosas, and aplorogramma. This was to further clarify the identification of those 4 species. We also did genetic diversity, and I'm going just to present on one of the species. Just as I said, we identified the 4 species, Numaeri was actually the most species, which we found it in most of the water bodies. So for genetic diversity in the species Apleurogramma, the sample size was 46, and we actually identified the species from the 3 water bodies within the North Rift, and it showed the haplotype diversity actually varied from 0.2 to 0.8, so the species showed high diversity. We also did a pairwise comparison of the species in Apleurogramma, and one of the water bodies, that is Cases Reservoir, showed high pairwise comparison of 0.4, and actually that water body had more haplotypes. It showed high diversity in that water body compared to Chebara Reservoir, whose was lower of 0.1. We also did minimum spanning network on Apleurogramma, and haplotype 1 actually was found in the three water bodies, that is Cases, Capsina, and Chebara. So it actually showed that this species, which I'm going to present, Enteromias, which I'm presenting Apleurogramma, was actually showed high haplotype diversity. So that is what we found out. So from our research, we realized that CO1 marker indicated the 4 species, and we realized that it provides actually an accurate species identification, demonstrating its effectiveness in ecological studies. And as a reference, as you had seen during the first results, was ranged between 97 to 100, showing that it is a good, the barcoding, the CO1 was good for identification. The findings also suggested the potential barcoding is a good tool, and it will enable the relevant bodies after identification for conservation of unique species. Recommendation is that conservation strategies should actually incorporate genetic considerations, because this is a sure way of identifying these species to minimize the risk of inbreeding, and there should also be creation of protected areas of fish sanctuaries in regions that have high genetic diversity. There should also be continual monitoring of the Enteromias population to track changes that could happen over time, and this should be through collaborative efforts among researchers, conservation organizations, and local communities, so that this should be continuous, the data collection and research. Further studies also should conduct in-depth studies on these species in small water bodies, because we did sampling, and this should be continuous. So thank you for the presentation. I acknowledge NRF and ChemFree for their assistance during the research. Thank you, and may God bless you.
Thank you. You don't go, because we are inviting 2 or so questions. 2 or so questions. Yes.
Thank you for the presentation. I'm not sure if you mentioned it, but can your species hybridize, or are we certain that they are always distinct?
Maybe she can take note and then she can respond. Then we take more questions. Just take it down. Any other question, comment? All right, you can respond to that.
Yeah, I think, thank you for that question, that could the species be hybridizing? So I think we didn't go in depth to hybridization, and yeah, if further studies should be done on hybridization of the same. Yes.
Yeah. So related to that, Prof. Yeah, I think there have been some studies in the Mount Kenya region, and there was some element of hybridization among several Andromia species of that region. So it just calls for more detailed analysis and more widespread sampling, and using even more informative markers. Yeah. Thank you.
Thank you. Let's clap for Hilda as she sits. Yeah. Our next presentation is by Blasio. Blasio, welcome. So on the session of conservation, biodiversity, and conservation, our last presentation is by Blasio. Welcome.
Thank you so much. My name is Blasio Kupambale from Uganda, and in particular, Bositema University. I am a PhD student working on mosquitoes as arboviral vectors. So mine is still an idea. I haven't collected data yet. I haven't gone to the field, but I thought I should share with colleagues on what I intend to do, and probably if there's 1 or 2, 3 inputs, I can incorporate them to make sure I have a better study. So feel free to engage with the presentation. If there are any areas we feel we can improve a little bit, it will be fine with me. It is about to come. Yeah, let's be patient a little bit. And I don't think I will take much, since so that is the title. It is the influence of terrestrial and wetland ecosystems on species occurrences of Aedes and Culex mosquitoes as vectors of arboviruses, basically in northern Uganda. On the introduction, I'm particularly showing the intensity of arboviruses, starting with the 3,400 mosquito species that have been identified worldwide, and then drawing down to the Culex and Aedes with the several species. And we know that these are infectious pathogens that in most cases affect humans. So I dropped down to showing the global scale on which countries have a high intensity or high number of mosquito species. I also dropped down to Africa. Then I came to Uganda, trying to justify why probably this study is important. The problem statement is basically looking at the intensity of the danger caused by these arboviruses. We are looking at the 150 up to 200 patients being infected with the arboviruses, and they are hospitalized annually. Then away from that, when it comes to northern Uganda, there is quite such an outbreak that is greater than 24. That is with 7.5 prevalence among the population. And I got this from that gentleman. So he's talking of the chikungunya virus and the onyongo virus being estimated at 31% up to around 38. And this is serious when it comes to percentages. When you convert it to populations, you might be surprised by looking at the huge numbers that are being affected. But despite these statistics concerning the emerging infectious diseases and re-emerging infectious diseases in those areas that I've mentioned, very little has surely been done. We have only known how people have died, but studies have not gone ahead, or government has not investigated on the abundance and the ecological ecosystems that these mosquitoes could be occupying. So I thought I should venture into that. I close the gap so that I can easily tell that we have such and such a number of mosquitoes, either in this area, could be terrestrial, or maybe wetland. I have majorly 3 key objectives. The other one is the general objective. But the first objective will be looking at determining the occurrence and abundance of these species. Basically, I'll be focusing on the Aedes and Culex mosquitoes, and that will be in the selected ecosystems that I already mentioned. The second objective will look at the abundance and occurrences in the wetland ecosystems. Then the third objective will be looking at the prevalence of occurrences of these abviruses. Here we'll be doing a screening to check which species has which viruses, and probably what is the intensity. So the methodology, we look at, the study will look at 4 districts, that is Gulu, Kitgum, Arua, and then Adjumani. And it will be a cross-sectional design that will be conducted twice. That is considering the wet and dry season. And I've indicated April to August. By now, actually, I'm supposed to be in the field. I already made some preliminary visits to mark up the areas and looking at the manpower that I'll be using. Then for objective 1 and 2, I will look at 10 sites per district. And then this is where the traps will be laid to make sure I get the samples from these different ecosystems. Now somewhere in the larger document, I have defined what these ecosystems could be. The wetlands, someone might ask, what are these wetlands? Maybe if it comes to terrestrial, another person will ask, what are these terrestrial? But they've been defined at some point. So mosquito trapping and collection, mosquitoes will be collected. We'll use 2 types of mosquito traps, the BG and then the CDC light traps. At that point, we will use 20 of them, and of course these will be distributed. So these traps, while being laid in the field, they will be assigned unique identifiers. So we shall have 5 traps that will be deployed indoors, and more 5 will be around the wetlands. The other 5, and the 5 light traps, then the 5 BG will be strategically set on the drylands. So the outdoor collections, these all traps that will be set outside, we hope to set them at 4 p.m. at an interval of at least 100 meters. And basically this is to avoid interruptions from between the lights. So all traps will be suspended above the ground at about 1.5 meters. Then for the indoor traps, those are the CDC light traps. They will be set at around 5 p.m. That is towards the evening. And all traps, both indoor and outdoor, will be retrieved in the following morning, beginning with 8 to around 9. So we'll assign them GPS to make sure we ascertain where every trap has been set, and we can easily retrieve back if we wanted to do any other development about it. So the preservation and identification, we will have, we intend to have a mini lab in in the field where we can do some sorting after these mosquitoes have been brought from the field. Of course, they will be having a number of other arthropods and probably unnecessary specimen that we don't want. So all these will be handled at the field level so that all the mosquitoes will be sorted from those other things, and then labels will be included on these different mosquitoes and from the different area where they've been trapped. And of course that will look at the date, the site, and then the trap ID, and then the number of mosquitoes and the habitat type. Is it from terrestrial or is it from wetland? So we will then transport them. We'll first pack them into the cryovials, and these will be labeled while being packed in Ziploc bags, and they will be stored in ice to make sure we maintain some shape of those mosquitoes so that they do not go otherwise. So the cryovials will be then transported to the UVRI. I intend to do my lab work from UVRI, though I haven't harmonized with them, but that is the nearest that I can trust and do my work with. And at the laboratory, of course, specimen will be stored in an acceptable temperature, that is at -8 centigrades. And then after that, the sorting and storage will be maintained under the cold chain conditions. And this will be basically to preserve the DNA and the morphological features of the specimen. Away from that, the process now will have the identification, and of course this will also be done in the lab. So that is UVRI, the entomology lab, where mosquitoes will be retrieved in the freezer where they are kept. So these mosquitoes will be morphologically identified, and we will use a standard morphological keys. We are referring to Edwards. So that the identification will be done on a chill table where the sterile microscope using the phainosophets will be used. So these mosquitoes will be identified and will be carrying out speciation. At that level, we hope there could be damaged mosquitoes that will need probably more identification or more experts, but we'll also make sure we do some identification using different experts who are well versed with these insects. So these mosquitoes will be pulled in the 2 mils cryovials with about a maximum of 25 in each vial. And the pooling will base on the sex, the physiological status, then the species, is it Aedes, is it Tulex, then the trap type, the date, and where it was collected. So these, the cryovials will be labeled with the specimen name, sex, date, like I had already described them. And these cryovials containing the identified mosquitoes will be organized in cryo boxes, labeled, and then stored. So, and this will be preparing them for the next process. And this will be the screening for the viruses. And here we'll be using the real-time PCR, which will be used to determine the prevalence of these viruses in the collected mosquitoes. And we hope to use the chi-square test to evaluate the heterogeneity of the rates among different study sites. Data analysis, we look at the comparison of mosquitoes and the species counts for the mosquitoes captured across the selected areas. So we'll use that formula to calculate the number of mosquitoes in a particular place and comparing the numbers and the counts. Then we'll analyze it using, we hope to use the SPSS, and some descriptive statistics will be used to summarize the data. So the difference between the mean numbers of Aedes and Culex across the studies will also be analyzed or assessed using the two-way ANOVA, and the Spearman rank order correlation will be used. This study seeks actually to address the trend of virus species occurrences and abundance, and then the ecological distribution of mosquito species belonging to these two Aedes and Culex genera in northern Uganda. Thank you very much.
Thank you very much. We'll take a few questions and comments for Blasio. You can take notes, then when we are through, you can respond. No, just stay here because we are finalizing now.
Okay, thank you. Yeah, thank you, Blasio. So these viruses, I see you mentioned many viruses. Some of them are new to me. I'm not very conversant with them. So I've heard of chikungunya, which is also very common. I've seen that it's very common in Kenya, I think at the coast, and the others with some difficult names. So are these species specific, or there's cross infection across different mosquito species? That's one. Then number two, I don't know if you plan to include some social aspects. Yeah, social aspects to your study. I inquire on this because the region you've chosen to study, I think, is a nice place. I imagine that it suffers from heavy disease burden because of the location, the infrastructure, the fact that it's close to the border of South Sudan and Kenya. And we know the challenges that those border-based districts or counties, if it's Kenya, face. One thing is that there's insecurity. Another one is that there is frequent movement of people across borders because they have relatives this side and the other side. And whenever government intervenes in some cases, you find that people freely move to the other side to evade the intervention that is being brought to that place. So I don't know, maybe you might want to consider this, or I don't know. Yeah, thank you.
Thank you. Any other question or comment? Yes.
Yeah, thanks for the interesting presentation. I wonder, because you take a lot of effort in finding by molecular techniques the viruses, I wonder why you don't use the molecular techniques also for determining the species of the mosquito? Or is there no reference database, or wouldn't that be quicker than microscopically identifying?
Okay, have you written that? Oh, you repeat?
In short words, I see that you have on the one hand high-end molecular techniques to identify the viruses, while the mosquito species, if I got you correctly, will be identified with a microscope. And I wondered why you don't use the DNA techniques also to identify the mosquitoes.
You got that question? Okay.
Is he listening?
Are you listening, Blasio?
Okay. Yeah. Just two simple questions. First would be, why did you choose the 4 sampling sites or the 4 districts? Like, what is special about them in terms of mosquito ecology? And the other is even simpler. If you focus on Culex and Aedes, what do you do with the rest of the mosquito species that you will capture in the traps?
Is that you're okay with those questions? We can respond to those ones first. Respond to those 3 questions, please.
Yeah, I very well know that this is an interesting area, and I probably wish to call the audience that you need to pose a few questions. I'm still loading. When I come for the results and the rest, then we will enjoy the thing much better. But to begin with, the first question was about the virus specific to the mosquito species. Yes, there are those that are specific to specific mosquito species, and of course most of them are so common with these 2 genera of Aedes and Culex, and this is the reason why I decided to pick on those, so that I could surely exhaust that area. Then the other question is about the social aspect. For now, we've not looked at this. I am using we. Of course, we know that every student has a number of supervisors, and they are all part of the study. So we haven't yet thought of this, unless maybe I table it to them and we see the possibilities of having it on board. The key-night technique, I'll also take note of this and share with my supervisors if we can, if it is possible to use it. The only challenge is that maybe these other techniques might be having other costs associated to them. One presenter here did highlight the issue of funding, and he was blaming himself that people of his color may not hear it well when you say there's shortage of funding. But for me, since I am a black man, funding is a challenge, and I'm okay to say it out. Yeah, so we may not do that. Then there is one concern about what is special about the districts. Of course, these districts are almost at the border, and other studies have been highlighting a number of infections that are happening in these districts. So it is those are some of the reasons, but also given their geographical setup, they are quite interesting that we may need to understand why we have these other strange diseases. Yeah. Without forgetting that, of course, the government has not ventured much into these areas. You will agree with me that it's very hard to cross to a different country when you do not have a yellow fever card. But no one will want to know if you have malaria. You can cross at any time with your malaria, with your everything. But yellow fever, everyone needs to know, and you can't get that card for free. So it is an area that we need to find out. If we can pay for yellow fever, can't there be something to be done with these mosquitoes that are responsible for it? Thank you so much.
Thank you. Is there any more question? Yes, there is one more.
I had already advised that they wait for results, but it's fine.
Not really a question, just a suggestion. I just wanted to ask, thank you for your presentation, by the way. I just wanted to ask if you have access to other microscopy facilities like scan electron microscope. So instead of relying on binoculars to identify your species, you use scan electron microscope to scan the mosquitoes or whatever you will collect and analyze those data, and then I think maybe it will give you a more or less accurate result than only basing your analysis on binoculars. Thank you.
Okay, thank you very much. I have noted that with concern, and actually very soon I will be heading to the Ministry of health in Uganda. And those are some of the issues that maybe I will discuss with them if I can get those equipments to help me do a better job. Thank you so much.
Thank you very much for those nice presentations. Let's clap for Blasio as he sits. Thank you so much, and welcome again to Kenya. We can wrap it up at on the sub-theme of conservation, biodiversity and conservation. And I want to thank all of you for your patience and allowing the presenters to present. Those who are presenting and they are yet to complete their work, we wish you all the best. Take note of the comments and suggestions. Stay along because this is a symposium and you learn a lot in terms of molecular techniques that you can employ in your research and that you can improve your publications and your research. Thank you. I want to usher in the next sub-theme, and our next sub-theme is genomics in animal breeding. The chair of the session will be Dr. James Barasa. Welcome.
Yeah, so thank you, Emily. So we move on to the next sub-theme, genomics in animal breeding. And without taking much time, may I welcome Professor Raphael Murode. I think he would be making an online presentation. Yeah, Prof, I'm not sure if you've logged in.
I'm here with you.
Yes, yeah, thank you. Yeah, so Professor Raphael Murode is an established figure, an established scientist in animal breeding or livestock improvement. He has developed a lot of models to support animal breeding, milk production, and important traits. He's a professor at the University of Edinburgh. There's the Scotland Rural College where he's based, but he's also a researcher or a scientist at the ILRI, International Livestock Research Institute in Nairobi, where he supports a lot of research work on animal breeding, especially on dairy, goats, sheep, and such. So he's well-versed in models, and so we welcome him to give his keynote speech on animal breeding, and we hope that we will really tap into your expertise to help us get some of these models. Thank you, Prof. Feel welcome.
Thank you very much. I just want to thank the organizers for inviting me to give this presentation. I do apologize that I couldn't be there in person, but I really count it a privilege. So I will share my screen and so we can get on with this topic of looking. Let me just put it on presentation mode. Give me a second. So I'm sure you can see my screen now. Okay. So thank you very much. So we are going to be looking at genomics as a pathway for efficient breeding programs in dairy system in lower to medium income countries. And as I've been introduced, I work at Edinburgh, but also I spent a sizable proportion of my time at Erie, Nairobi, Kenya. So I thought that initially it would be very important before we look at genomics as a pathway to an efficient genetic improvement program in dairy is to initially actually define what is an efficient and a sustainable dairy genetic program look like. the components of an efficient and sustainable dairy genetic improvement program will include these vital, very vital components. There will be improvement in productivity. That implies that it is actually economically viable. The farmer can actually see the benefit of such improvement program in terms of income and improvement in livelihood. We expect such a breeding program to actually improve fitness. You are not just improving productivity, but you want the animals to improve in terms of fitness, reproductive ability, disease resistance, and robustness as well. We also expect such a program to actually improve welfare traits. We want the body condition of those animals to be good. We want those animals to have sound feet, and so on and so forth. And finally, and also very important, given the issues we're dealing with in terms of climate change, we expect that there will be little on no, on very little or no negative impact on the environment. So these are the components of what we actually would expect from a very sustainable and efficient dairy improvement program. And the question is, why do we think genetic improvement is an important component in a breeding strategy for dairy? The reason that genetic improvement is a very important driver in any system in terms of a breeding strategy to improve productivity in dairy or in any other livestock. The reason is that the results are permanent and cumulative. It means once you achieve a genetic change, it is there permanently in as much as that population is maintained. We have more product per animal, so few animals will be required. That implies that the impact in terms of the environment is reduced. And of course, related to the first point is that the genetic solutions you provide in terms of animal health or welfare require less labor or material input than chemical or mechanical approaches. I mean, every year you don't have to spray the animals, for instance, if you can improve them for disease resistance. So in a sense, on the long term, it's an efficient approach in terms of achieving an improvement in productivity, either in health or in yield. The question is what actually does genetic improvement look like? And so here is just basically the very principle that drives genetic improvement. And it is important we understand this. And when we do so, we cannot begin to understand what role can genomics actually play. So if we start with any population, usually there's a normal distribution until we have the top producing animals and we have the lower producing animals. The role of genetic improvement is to identify these top producing animals and to use these animals as the parents of the next generation. And if we're able to do that, what will happen at the offspring of this parent generation, we perform better. So this is the mean in the parent generation. Now in the progeny or the offspring, we find that the mean has increased. So that difference between the mean of the parents and the is what we actually regard genetic improvement. But what actually drives it is our ability to identify these animals accurately and in an efficient manner. This is the role that genomics can actually help us to do. But prior to genomics, what has happened? Prior to genomics, what will tend to happen that whole process of selecting animals will be based on pedigree and the actual records of performance, like milk yield or body weight. And analyzing those and applying models, we have actually seen that it has resulted in permanent increases in productivity. So for instance, this is the increase in milk yield in developed countries, starting right from 1998 and so on. So we can see that there's this upward trend in productivity from genetic improvement, selecting the best animals to become parents of the next generation. And however, when genomics came, that took another dimension. For instance, this is the UK dairy breeding. This is a very brief snapshot. We introduced genomics in 2012, and you can see that from there that there has been a very increase in terms of the proportion of young sires that have been used. So we are using young sires now. Prior to this, before you use a particular bull, you're looking at both progeny from 5 years, 7 years old bull. But now we are using boosts at the age of 2 to 3 years old. The implication of that is that we have actually doubled the rate of genetic progress. And so if, for instance, we look at this graph. This graph is just basically telling us, for instance, what happens the percent to the progress that is made in several farms, depending on the proportion of animals that are derived from genomics. And of course, the top one, which is the blue one, we have farms that have more than 75% of the cows derived from genomics, genomic breeding. And the very last graph here is for cows that have less than 25%. You can see that the massive difference in terms of the progress they are making. And so that illustrates for us the role that genomics can actually play. But the question is now, let us not really look at low or medium income countries. Where are we really? And what role can genomics actually play? So first of all, we review the current dairy system in low to medium income countries. And what opportunities then do genomics actually offer us to drive genetic progress? The reality of the situation and the breeding program based on selection of superior animals are generally lacking. When I say this, I really mean selection of superior animals, not importing from abroad. We have not a system whereby we evaluate our animals, we select the best ones, and we use that for breeding the next generation. That is real permanent genetic progress. This is generally very lacking. Whether in the dairy, we have a bit in this small ruminants in some countries like Ethiopia, where we have the community breeding program, but in general, such breeding programs are lacking. And then we come to the issue, we have limited data infrastructure. So systems to actually collect routine data and pedigree are generally lacking. Our data set tend to be small because we have small farms and small progeny sizes. So the sires that have very small sizes of progeny. Limited herd sizes with animal effect confounded with herd effect. What this means, for instance, we have this smallholder farm that produce 80% of milk, for instance, in Kenya. They come from smallholder farm. They have 2 cows, 1 cow. And what happens is that when you have 1 head and 1 cow, you cannot separate the effect of genetics from the herd effect because 1 cow and 1 head, they are confounded. And above all, we have unplanned crossbreeding. We don't know the parents of these crossbred animals. We don't know the breed proportion. We cannot say this animal is 50% Holstein or Jersey or indigenous breed. And of course, in a heavily reliance on importation. And this importation is like dipping our hands into a black box and just choosing an animal. We have no way to actually assess whether the animal you're actually importing will actually perform well under the smallholder system. So these are the limitations. Now the next question is, how can genomics help us to address some of these concerns and therefore enable us to actually drive genetic progress? The first one I would like to talk about is this inadequate data structure. Where we have lack of pedigree, our sires have very small size of progeny. How can genomics help us then? What happens in genomics that we take hair samples or blood samples and we genotype these animals? And that means we have SNP information, genotype information. This enables us to construct a genomic relationship among the animals. And once we're able to do that, because we don't need the pedigree, because that is the main function of a pedigree, once we're able to compute that information from the genotype, it means there's less reliance on pedigree. And that means we can actually analyze that data and begin to select the best animals to become the parents of the next generation. So that is one of the ways that genomics can actually help us. The other one is this issue of limited head sizes, whereby you have a few animals in a head, so the effects are confounded. What happens is that because of genomics, because the sires are used across heads, the genomic information from these sires enable us to tie all the heads together. So we use some terms like the haplotype blocks in the gene matrix, which is shared among the common cells used across there, enable us to separate the effect of the head from the animal effect. And I'm just going to give you an example of what we did. We've done this in several African countries, but I'm going to use the example of Tanzania, where with the African Dairy Genetic Progress, which is at IRI, funded by Bill Gates Foundation, has been working for several years, and including other countries like Uganda, Kenya, Ethiopia, and so on and so forth. When we look at the Tanzania data, you can just see what happened. We have here the pedigree. We have 88% of those animals, we don't even know their parents. For 11.4%, we know just one parent, and it's only 0.6 that we know both parents. So you can see that this is so sparse, and there's no way we can actually use that information to actually select, to estimate the breeding values and select the best animals. And moreover, 50% of those cows were in one herd, so we cannot separate the effects of the animal from the herd. And so what we did, we genotyped around 2,000 animals. Because we have that genomic information, we're able to compute the genetic relationship, and so we can do prediction, what we, in genetic terms, we call it G-block, or we can actually, even in more better way, combine that genomic information with the few pedigree we have and do single step. That means animals which are not genotyped. For instance, we only genotype 2,000 animals. We don't more than that now. But it means that all the animals in the pedigree, we actually benefit from that information, and that's what we call the single step. We combine the pedigree and the genotype together. So when we do that, this, for instance, just an example of what we estimated. So for instance, we're looking at milk yield. We can actually estimate heritability of 0.1, so that the degree to which milk yield is controlled by genes. For both 0.24. So these are very reasonable breeding values that will enable us to select the top animals to become the parents of the next generation for milk yield or for body weight. But more interestingly, this is actually the most interesting bit of the work that genomics gives us. This gives us information that we can actually predict the genomic ability of an animal to produce milk or body weight at birth. All we need to do is, for instance, take a hair sample, a blood sample, and once we get the genomic information, we cannot predict that this cow, even though it's just 1 year old, this is the milk it will produce with this amount of accuracy. This means that we can reduce generation interval. That means we can select the best animals at a very young age and use those animals to become parents of the next generation. And really, this is what drives genomics, reduction in generation interval and our ability to select animals at a very young age. And as I said, we've done this in several countries, and what we actually produce, we now begin to produce information that will support such genetic improvement. So we can produce, we have a bull and cow catalog in Ethiopia, we have one as well in Tanzania, and we are just working on the one for Ethiopia, for Kenya. It means that farmers now have access to this catalog and can determine which bulls actually are the ones on top of the scale in terms of productivity and so on and so forth. So you can see how genomics have helped us to overcome that limited data structure. And I've just put some graphs here to see when we started the program in Tanzania, the average milk yield in the farms we're working was around 6.75. But now we're looking at average of around 10%, which is more or less very close to almost double what we started with. The same thing we start with Ethiopia. We started from around 10% because we're using a subset of farms, but we have included more farms. So initially there was a decrease because we included some poorer performing farms, but we begin to see an upward trend in the performance of milk yield in the farms we're working with. I wanted to include the one for Kenya, but this is restricted to small farm and medium farms in Kenya. You can see the upward trend as well in year from around 6 to now 10 kilograms per day. And so we see that genomics actually helps us to overcome some of the limitations that are existent in our current dairy system and enable to begin to drive genetic progress. Another problem which I also mentioned to you is that we have crossbreeding, and it's just random. It's not planned, and the farmer just use a particular bull and so on and so forth. And and we don't know. You look at a calf, we have no idea what is the percentage of Holstein or Jersey and so on and so forth. You may ask, why is that important? That is very important because we need farmers are at different levels of production. They have different production systems. Some have not a lot of resources, but have very limited resources. And if you have limited resources, we don't want you to have a cow that is like 80% Holstein because you do not have the resources to feed such an animal. So what we can do, what genomic help us to do, I've seen some of your presentation, we can develop a little SNP from the SNP information we have. So for instance, using the 50K or the high density SNP chip, we can find the proportion of the different genes in that animal, but that is rather expensive. So we want to say, can we reduce the number of SNP? And you find that 200, you almost get the same accuracy that you get using the whole high density chip. The consequence of this is that we have developed some chips of lower density, 200 SNPs, that enable us to determine, for instance, this is the proportion of foreign genomics and the indigenous breed in the particular animal. And if you want to know the parentage as well, we need to increase that number of SNP to 400. We have improved on this. Now we can actually separate Holstein from Jersey in an animal. This is the percentage of Jersey, the percentage of Holstein. As I said, the importance of this is enable us to select the genotype for the relevant production system. So if a farmer has a lot of resources, then you can give that farmer or recommend for that farmer an animal with a bit higher percentage of exotic gene. And then we come to the issue how we actually utilize our indigenous breeds. Our indigenous breeds represent a unique set of genotypes. You know, they survive under harsh conditions over many years and are very resistant to disease and parasite. And this is very particularly important, especially given climate change. And what genomics offers us is enables us to understand why are these animals able to survive in a harsh environment, or how are they able to show tolerance to a particular disease. And genomics gives us the ability to do so. We can do sequence of selection, or we can do GWAS, and we can actually isolate and identify the genomic region that confer tolerance. And so Tijani, who works in Erie, was able to identify some region that confer troponin tolerance. We work with a group in South Korea, and we're working with 12 African dairy cattle, and we're able to identify regions that are related to immunity, heat tolerance, tolerance, and the reproduction-related traits. So we can now know that this particular region enables these indigenous breeds, for instance, to survive under these hard conditions. Now, the next question is, how do you use that information? The situation is that when you buy the current chip for genotyping, they were all put together using European breeds. And so they lack all the information. They can only explain 83 to 90%. So when we have genomics, you can actually put together a functional SNP, a customized SNP that have functional areas and genes that actually incorporate some of these regions that we're talking about. So you have animals that if you do selection, you want to include those regions of the genomics to make sure that you don't lose the ability to be adaptable to the local conditions. And once you're able to do that, then it means that you can use your customized shift to select animals that are both productive and also adaptable. The next one that we can also use, there are various methods we can use to use that information. And one of the papers recently published called GWAS blob is means that you use GWAS, and you can use the result of those GWAS to weight your analysis and incorporate those information to your analysis. And it drives accuracy up by almost 10 to 21%. And then the other one is that, because we have genomics, we can derive, and I've been hearing the conservation, you can actually design the conservation through sustainable use of these breeds. You can do a PCR analysis to understand the population structure, the genetic diversity, and you only want to conserve those very distinct genetic resources. And in terms of cattle, because for the local breeds, the population are not very huge, we need to control inbreeding. And genomics enables us to do that. We can compute inbreeding from the runs of homozygosity. All that information comes from the SNP or genotype information. However, we find that the two factors that actually drive genetic progress or genetic improvement when you're looking at genomics is the accuracy of genomic prediction and how intense you're able to select those animals. And these benefits are amplified, or we get more if we can combine genomics with assisted reproductive technologies like AR or IVF or ET. It results in a reduction in the generation interval and means that we can select more of the top animals and use the genetically superior parents in a better way. So there is a project we call the COOCAR project at SRC. And the purpose of this project is, of course, we know that climate change is upon us, everybody, and we want to reduce the amount of methane that is produced so that we do not have a major impact on climate change. So the idea was here we'll have genomically selected superior bulls. So these are bulls that be genetically selected that we know they produce low methane. But how do you ensure that you can actually roll that out in a quicker way so we can fertilize eggs using the semen for the superior bulls, and then we can do embryo transfer into surrogate dams. By just doing that process, you have reduced the generation interval by around 8 months. And that means you can rapidly spread the effect of these superior bulls that reduce methane across the whole population. So this combined use of genomics and reproductive technology actually implies that we can actually drive genetic progress at a more rapid rate. And of course, we all have heard about gene editing, which allow animal breeders to presently add or delete or replace letters in genetic code that difference a particular trait of interest in just one generation. This company, Assygen, actually have sire gene lines which are already be edited for heat tolerance, sleek gene, and other traits. So when we combine that information with reproductive technology, it means again we can actually rapidly multiply the effect of such genetic manipulation from genomics. So we have the surrogate sire technology. So basically, if you combine the surrogate sire technology with gene editing, it's got another very good potential for the rapid dissemination of the superior genomics that come from genomics. So basically, what the surrogate sire technology does is to create maize that lack their own germline or germline cells, and then you transplant this mitogenous stem cell from the donor or the edited male that is genetically superior. So by doing that, the single elite male donor with this novel trait or genomically selected can give rise to a huge number of progeny. And so by combining genomics and reproductive technology, you actually rapidly disseminate the superior genomics that we derive from our genomic application of genomics. And the reality of the situation is that most African countries, most low to medium countries, they still continue to import foreign bulls. And as I said before, this whole process is like putting your hand in a black box and just picking something out of the black box without knowing will this bull actually do what is saying we do. Of course, these bulls, when you buy them from abroad, they will have indices abroad. But these are systems that are based on high production systems, high input, and bringing such animals to the low to medium income countries where feed is restricted, is limited, and so on and so forth. It really becomes a challenge to those animals in terms of their performance under the smallholder system. And so the question is, with this, can we use genomics to actually help us when we are selecting animals that really we actually be able to bring in the animals that actually will do well in the smaller holder system? So this is the question: can genomic information be used to profile suitability of imported animals for making in smallholders in East Africa. So we did some work on this, and basically we did it with the UK. So basically we have the bulls that are marketed in the UK. So for instance, most countries, when they go to Africa, that's what they will go to the bulls that are marketed, and they will bring in those bulls. So we have those bulls that are marketed. The what we did, we have the SNP effects, how the bulls actually perform in Ethiopia and Tanzania, and we computed what the breeding values, how they actually perform in Tanzania and Ethiopia. And then we now have 3 pieces of information. We can now rank them using these 3 pieces of information and say, how would they actually have performed if we have used that information? You know, we find that if we select these animals based on their performance GBV abroad, actually those animals, when they are brought to Africa or East Africa, they show average or very poor performance in both countries. They were never top performing. However, when we use the information we generated from genomics for these 2 countries, we find that it resulted in performance up to 20 to 35% standard deviation above the average animal. The question we're asking is, how does these animals, or what sort of categories of these animals were in the exporting country? We found that these animals were actually often average performing animals in the foreign country. So we find that we may not likely buy them because on average, they are just average in the exporting country. So this shows that genomic information, we actually allow to select foreign bulls that will do better here in our smaller holder system. We're actually working more on this to take in more than milk and to include other traits as well. So the question that comes is that we can all see the benefit of genomics, but the usage is limited. So what are the challenges that hinder widespread use of genomics? The first thing which you may have recorded that is the lack of this systematic way to record data. It is very fundamental. As one of my colleagues says, in the age of genomics, phenotype is still king. The reality that if you cannot measure it, you cannot improve it. In any system, whether any business, data is fundamental to drive such system. And so we must find ways to collect data. So we have tried at ERI several ways to actually do that, digital tools. So we have the ODK on tablets. Now we have developed a smartphone that enables data to be collected. We have also farmer-based systems which are suitable for USSD phones in case they don't have smartphones. Again, these are very useful. And India, they have several forms of data collection as well. And I know the community breeding program in Ethiopia, they have a system, a cloud-based to collect information. And I really want to encourage us to be able to explore digital means to collect data. It is much easier now. It's just as simple as using a PESA to transfer money. You collect the data and it's remotely transferred to a database. And it's something that I really want to encourage all of us to actually be involved in. And of course, we can use sensors as well. The colors that are worn by the animal to collect very novel phenotype from fertility. Another way in which we can see now, if I look at the animals that are genotyped from some I studied that these are from projects which are development projects, they're breed societies, some cases in Brazil. We don't have AI companies driving these processes. So we still have to send our samples abroad for genotyping. So we need an approach to reduce the cost of genotyping. And so genotyping animals that jointly services many of beneficiaries, such as research institutes, university, and farmer cooperative, and individual farmer is very beneficial. So when you genotype an animal, make sure that animal will serve several purposes. It can be used for research, for training students, can promote the health, it can deliver benefit to the farmer. Therefore, the different organizations can jointly contribute to the cost of such genotyping, and that will help to reduce the cost. And of course, from the examples I've given, if you combine it with reproductive technology, the cost comes down. If you use sex semen, for instance, or various reproductive technologies, IVF, ET, and so on and so forth, the cost of genomics, actual genotyping comes down. And so I'm going to be concluding, genomics offers the opportunity to actually improve efficiency and suitability of the dairy system in developing countries. We can do genomic prediction and select, even we have limited pedigree information. Implied breeding program can't be commenced now. You can actually start using genomic selection and select animals of superior genomics that can be selected at a very young age. We can use genomics to actually tell us the proportion of genes in that animal. So we can target animals for various production systems, and we can discover what the parentage are. It offers the opportunity to model underlying genetics for resilience. We can understand why this animal shows resilience or is adapted to different health conditions. We know that reportation continues to be part of the dairy count improvement, and genomic will help also develop a suitability index. So when we are importing animals, we actually import those ones which are relevant and we do well in our countries. And of course, strategic genotype is important so that we can reduce the cost of genomics. Thank you very much. So I want to acknowledge this project, as I said, was funded by the Gates Foundation and my colleagues here who work at ERI on the whole project in several countries. And of course, I often say the dairy farmers and the various research organizations in different countries that support the activities that we do across these several countries. So thank you very much, and I look forward to your questions and comments. Thank you.
The comments? Yeah, you are welcome.
Yeah. Thank you, Prof, for your good presentation. This is a bit general. I'd like to know your view now with way genomics is also, as you've talked about, the role in animal breeding and genome editing. So what's your view regarding, genome editing, its regulation, and where we could be maybe in a couple of years in terms of animal breeding with all these tools that are coming, and maybe talk about genetically modified organisms in terms of animals and where we are also headed with that. Thank you.
Okay, thank you very much. So I think the first debate has been the question that was the main issue of debate was, first of all, is gene editing and GMO the same? And that has been taking place in several countries, developed countries, developing countries, and different countries have adopted different strategies. But just to really indicate, first of all, gene editing is basically the idea is, as people will say, it's like clipping, is deleting a particular section maybe of the genome, maybe because the animal is double recessive with a particular trait, so you change that bit. So in a sense, you're deleting without introducing any foreign gene. And so I think when editing is done without introduction of any foreign information, I think generally most countries are open to that, even some countries in Europe. And I know particularly for Kenya, because we're working with bringing some gene editing animals, that the government is open to accept those animals in as much as there is no insertion of foreign genes or genomic or genetic information from another organism. So I think generally it will play an important role in terms of especially disease resistance. We have some really challenging disease issues in livestock. ACF, for instance, the swine fever, ECF for dairy cattle. And I think there's a lot of work trying to see, is there any way we can actually do gene editing to actually address some of these major disease issues? And for me, I really feel that gene editing will play a major role, especially a very relevant role, if our focus is really on addressing some of these major disease issues. Now, when we come to GMO, it's a real challenge because it's involved introducing foreign genetics into an organisms, and at times evaluating the impact of that insertion is uncertain. And I think most countries, and even I know Kenya, is GMO is a no-no. And I think even in Europe is also a no-no as well. I think America is a bit more liberal in that respect. So I think the way I look at it, I can feel that gene editing will be important, not necessarily maybe for it might result in improvement or productivity, but I think for health rates, for disease as well, it's going to be important. Another area that we focus upon is this heat tolerance. So basically the idea, for instance, that, okay, we bring Holstein or foreign genomics or genetics into Africa, one of the major issues that challenge them is the heat. So if, for instance, we can actually edit these animals so that this heat issue is actually taken care of, so that there's high productivity and yet adapted to the high temperature, to me, I could see the benefit of that. And I think I see that in the future, I can see gene editing playing an important role for disease, for issues of adaptability in terms of heat tolerance and so on, given climate change. For GMO, it's always be a controversial issue because we cannot fully evaluate the impact of such a change. Thank you very much.
Yeah. So the next presentation is given by Professor Hans Michael. So before is just set. Yeah, so Professor Hans Michael is a professor at the University of KwaZulu-Natal in Durban, South Africa. He works on honeybees. Yeah, he works on honeybees. So when you see honey or when you buy honey, it probably has come from some of his efforts and studies and all that. But I know that he has also worked in Nairobi, in Kenya, stayed in Kenya. He used to work at the ECP, I think, yeah. And ECP used to have a very large program or project on honeybees. And he was instrumental in establishing the project. And after that, he moved to Durban, the University of KwaZulu-Natal. So welcome, Prof.
Yeah, thank you. Yeah. Thanks for that introduction and for giving me the opportunity to tell you something about bees so that you can learn a bit about these wonderful creatures. And this is still some work that was actually done in Kenya when I was working here. Most of that work is by Zamira, and that was for her MSc thesis. And you made also a good point. You were immediately saying bees, the honey and the honey, the honey. So that is what most people do. But I think there's also another important thing, and that is pollination. And this is a picture that shows you a supermarket. On the upper picture, you see all the produce with respect to fruits and vegetables that are there. And on the lower one, everything that is dependent on pollination is removed. So it shows us that pollination is certainly important for food production, and indeed 75% of all our crops that we use for our consumption are dependent on animal-mediated pollination. And that makes an overall global value every year of 150 to 500 billion euro globally. So that's a huge amount of money, so it depends on how you calculate that. And that makes honeybees the third most important livestock animal on Earth, after cattle and pigs, but more important than chicken. Good, so that's important, but we also know that bees are, it's not working. I can also use this. So it's also known that bees might be declining, and for example, the Time magazine had a cover in 2015, a world without bees, and everything is black. So a world without bees means that there are no flowers left and so on, because they are dependent on these pollination services. And in addition, we know that there are certain factors that drive these pollinator declines. So this is from a study that was recently published, where some experts looked at the different continents. What are the factors driving pollinator decline? And when we look at Africa, there are three major things popping up. So the larger the circle, the more important the factor, the darker the color, the more established it is that this factor contributes to the decline. And we have there land configuration and land use. That is one important thing. So the habitats that are lost. There is pesticide use in agriculture, and there's climate change. These are the three important factors in Africa. Good, so we were interested in the pesticides, one of those that is not well established, as you see, and pesticides, that is a cover for many different things, like insecticides and acaricides and herbicides and fungicides and rodenticides. So it depends on what we want to address, or what the chemicals should target. And so some of them are very clear, like insecticides, yeah, they kill insects. So they are harmful to bees. So that was not so much our interest. We were more interested in herbicides, because they're also frequently used. But herbicides are actually made against plants. Are they also detrimental for bees? And we used two of them. There's one that's called paraquat, that is the chemical, and the company that, I will not name the company now, but they sell it under this name, Gramoxine. And there's another one that is probably even more known, glyphosate, or sold as Roundup, yeah, that is discussed widely. It's banned in Europe because of its detrimental effect to bees. So, but how can it affect bees when it's an herbicide? So what's the mode of action? Paraquat generates free radicals. So that means it causes cell stress, and that is what finally leads to death. And glyphosate, that interferes with a plant and bacterial-specific pathway by inhibiting this enzyme. I will not read it out now. It's EPSP. So it's inhibiting that, and what's the major effect of it? So the organism cannot any longer produce aromatic amino acids. That means it will also die, because they are important for our metabolism, or for the metabolism of the plants and bacteria. Okay, so this is what we wanted to use, and we know from a previous study that we did that paraquat and glyphosate have an impact on the survival of bees. So these are lab experiments. We have groups of bees, and we feed them different concentrations of these substances. The green line is always the control. And they survive quite well. And then we see in the dose-dependent manner, so the darker the color gets, the higher the concentration, that they quite quickly die at this concentration of 2 milligram per milliliter paraquat. And they also quickly die here with 8 milligram per milliliter of glyphosate. So these substances are killing bees. So now we want to understand, how can that happen? And the idea is that glyphosate, for example, targets a pathway that occurs in bacteria to look at the microbiome of bees. So bees have a digestive system that consists of several parts. There's a crop, that is where the nectar is stored, that then is later on put into the cells and transformed into honey. And then we have the midgut, the ileum, and the rectum. And we find there specific types of bacteria. It's a rather simplistic microbiome that bees have, that is shown here. There are 9 major taxa of bacteria that contribute to this microbiome. The one in the ileum is different from the one in the rectum. And these can be studied, because it's quite simplistic. They're also especially in the ileum, there are some species that are extremely bee-specific, and you rarely find them in other insects. Good. So we aim to investigate the effects of these herbicides, especially of acute and chronic feeding on the members of the gut microbiome, and we wanted to test then whether these members of the gut microbiome can also develop a kind of resistance against these herbicides, and we wanted to evaluate changes in the gut microbiome composition under permanent herbicide exposure. So how did we do this? This is for the first objective, our first experiment that we did in the lab, and here comes a point that is quite helpful. Bees that are hatching freshly, they have no bacteria in their gut, they're free of bacteria. So that makes it experimentally quite nice. We take these freshly hatched bees, put them in the cages, and then we can infect them with a microbiome of our choice. So we use these bees, for example, now to infect them with a microbiome from a hive environment so that they have a natural microbiome. And then we gave them an acute, that means once, a pesticide dosage that was fed to them, or a chronic that was over several days. And the acute one was either sublethal or lethal. Lethal means the LD50, so 50% of the bees would die, and the chronic was the lethal, just over 5 days. So that's what we fed them, and then we took samples after 5 and after 10 days, 10 bees out of these cages. They had 3 replicates. We had controls, a positive control. They had a microbiome, but they were not fed the herbicides, and we had a negative control. These were bees without any microbiome. We took the samples, and then we used RT-PCR to check for the abundance of 5 specific taxa of bacteria. Yeah? So the first 3 are in the ileum, and the second last in the rectum. So this is what we found. This is for paraquat. This is the acute feeding. There's always the negative control, the positive control, then the lethal, and then the sublethal concentration on day 5 and on day 10 for the different types of bacteria. And what we see quite clearly is there is a reduction in those bacteria. Yeah? So when you compare with the positive control, we see in all cases that there is a significant reduction. It is for that group, the Lactobacillus firmicutes 5 that occurs in the rectum is the one that shows the least reduction. Yeah? But all the others, some of them are quite strongly affected and reduced. So that means these bacteria can also not deliver their positive effects. Under the chronic feeding, it looks quite similar. There's only one treatment with chronic here. You see it's reduced for these specific ileum bacteria, and also here in the rectum, they're reduced. So this is the glyphosate, and it looks fairly similar, so there's a huge reduction in all our treatments. Again, we see for the Lactobacillus firmicutes 5, it's the least strong reduction, and if we look at the chronic one, then even here these firmicutes 5 are not affected, but all others are quite strongly affected. So that could be a way how herbicides work, because they reduce these microbiota that is actually essential also for the bees, because this has immune functions, detoxifying functions, and so on and so on. Good, but now we wanted to move on and understand whether these bacteria also can develop a type of resistance against these herbicides. And so how did we do this? This is now our next objective. So we did an experiment again with cages, freshly hatched bees. They have no microbiome. We can give them a microbiome of our choice, and then we feed them with either paraquat or glyphosate, or a combination of both, or we use controls that don't get any herbicides. And then we start now with our first cohort, we infect them with a microbiome from a beehive. That means in a cage, we introduce them into the beehive, and they pick up a microbiome there. It takes a day to establish, and then we can feed these bees in the cage with those different herbicides. So we do pesticide feeding, we monitor them until 80% have died in that cage. So we have only 20% survivors. And what we do with the survivors, we extract their microbiota, because that could be the one that contributes to survival. We extract it and put it into a new batch of bees. And we also count the time, how long do they survive until the 80% have died. So that's an indication of how well they are doing under a certain treatment. And we want also to see whether something improves. And then we do this again with the second cohort that has now this survivor microbiome, and we let them also die and take the 20% survivors, extract the microbiome, go to the next cohort and feed them the microbiome, feed them the herbicides, and do this again and again and again until we end up with cohort 16. So this is what we did. We took also from generation 1, 8, and 14 samples where we applied 16S RNA metagenomics to identify the bacterial taxa. We did this with the Oxford nanopore sequencing for all the 3 replicates. Good. So how does it look like? This is now a chart where we have the cohorts on the x-axis from 1 to 16, and now we have the surviving days until, so how long do they survive until we have 80% dead bees. Yeah? So this is our control here, and we have a median of 19.5 days. So that is fairly a good number. In laboratory experiments, usually the range of survival is between 20 and 30 days. So this is 80% of the bees lived until day 20. Yeah? Paraquat, we have a median of 6. So that's a huge reduction in survival. In the beginning, initially, the first 5 cohorts, they survived 3 days, and then 80% were dead. So it's really a deadly stuff that we fed them. So they recovered then a little bit and went up to the median of 6, but it stayed almost at that level. Glyphosate, again, the control on top to compare. Glyphosate is here. So here we see it's a slightly better than paraquat, but still, compared to the control, bad. Yeah, 8 days they survive. Initially, it was very bad. Yeah? So the first cohort, they could recover a bit then, and then they went up to these 8. But even that recovery was not reaching the levels of the control. Good, and if we combine it, it is a little bit in the middle. It was bad at the beginning, but overall they end up at surviving 7 days, much, much lower than our controls. So it affects them. We might be able to, when you look at this, there is a bit of a recovery. There is a bit of a recovery here. So it might be that there is a bit of an kind of an evolution of resistance in the bacteria, but it is not very strong, and it is not recovering to the levels of the control. Good. This is a summary of this. You see here the mean survival. We can calculate the hazard ratio for that, so how much a certain substance that we give them is a hazard to them. So you see this is between 8 and 10 times higher likelihood that they will die compared to the control. Yeah? And all of them are significantly lower than these controls. And now we looked at the microbiome. We sequenced the microbiome, as I told you, in our replicates at the different days, day 1, or cohorts, cohort 1, cohort 8, 14 for the controls, paraquat, glyphosate, and the combined treatment. That looks fairly similar like the usually the microbiome looks like. The composition is also fairly similar. There is something in the middle. This is light blue. Unfortunately, the contrast is not very good. I'm sorry for that. But there is something in the middle here. So the first thing that we see is paraquat. There's actually almost no diversity. So there's a huge reduction in the microbiome. So the substance almost kills almost everything, and there's only a very few left. These are the lactobacillus that we beforehand have seen. They can survive also in the acute and the chronic treatment. They are able to survive. It seems also here they are the ones that survive. In glyphosate, it's not as drastic, but there's also a huge reduction in the different taxa. There's also a lot of these lactobacillus that survive. And then, that's a little bit puzzling for us, in the combined treatment, it looks better than in the single ones. It is, so we have more diversity there, but it is, in comparison to the control, also a strong change in the microbiome. It's not a drastic reduction in the diversity, but it's a strong change. Certain taxa are amplified and others are reduced or are completely absent. And if we put this into, into a PCA, then we see that there is a clear clustering. So these are the controls. Here is the paraquat. That is very different from the controls, and these are the treatments that included glyphosate, so glyphosate alone or in combination with paraquat, so they cluster. So the communities of bacteria are very, very different in the treated ones compared from the control. And that could be a reason why these herbicides have an effect on bees. It is not the direct effect, it is the indirect effect, because they target the microbiome. Yeah? So we have seen there are negative effects on the members of the gut microbiome. Gut microbiomes might be able to develop a bit of resistance, but it is not very strong, and it cannot promote survival of at levels of the pristine bees. And changes in microbiome result in some kind of dysbiosis, also due to the elimination of certain bee-specific taxa of the microbiome. Finally, I would like to thank the beekeepers that helped with that work, and some organizations that provided some financial support, and I thank you for your attention.
Yeah, thank you, Prof. Yeah, so any comments or questions for Prof?
Yeah, that's really interesting. Yeah, and my question is, what could be the role of these microorganisms, in that you showed that when they are hatched, they are not there, but then over time they come, and then they become very beneficial, such that when herbicides are introduced in the gut, the bees die, or the highest proportion of them dies. So what is the role? That's the question number one. Question number two, is there a specific reason why lactobacillus seems to be resistant when these bees are given these two herbicides? They seem to be consistently high. It's like they're not being affected by the herbicide. Is there a particular reason for that?
Maybe I can start with this one. They
Yes.
live in the rectum. At least the two groups that we showed here, the lactobacillus, they live in the rectum, and that is like the last part of the digestive system. So that means the herbicides have to pass through, and they're not passing through with, unprocessed or something. Yeah, so because they are also detoxification systems. Yeah, so that the bees have. So that means what we start with is not what we end with and what they are exposed to. But in addition, because the two groups of lactobacilli also differ, It might be also that they have specific innate mechanisms to detoxify, to be resistant, whatever it is. But this is what we have not studied in here now. But would be maybe interesting to understand in the future. So is it just a spatial thing, or is it also something innate that they have mechanisms to deal with it? Yeah? So the first question was the microbiome, yes. So the one thing is when they hatch, they don't have it, because this is what happens during insects pupate, and during the pupation they shed out the everything, yeah, with their old cuticula, also the bacteria that were inside. And what in bees is happening, so they infect from the hive environment where they live. This is where they pick up the microbiome. So that is like the typical what is present inside of that specific beehive. This is what they use to reinfect themselves. And then it establishes inside of them, and then it has several functions, and the different bacteria have different functions. Some have, for example, a positive effect of the on the immune system, so they help with challenging other pathogenic bacteria or opportunistic bacteria that are coming in through the food, for example. Others help with detoxification. They have various functions. And this is even not fully explored. What are those functions? Yeah? Because in most cases, people look at single bacteria, and then we don't know how they also interact with each other.
Thank you. Any other? Yes.
Yeah, thank you, Mike, for your presentation. I don't know if I missed this during your presentation or the answers you have already given, but I was just wondering, in the 20% of bees that are surviving, so you are kind of attributing the survival rate to the microbiome, right? Yeah, so what about the contribution of the bees themselves, their genetics into this resistance? Yeah, and my second question is related to the microorganisms themselves. They have survived. You did some sequencing, but I don't know. What about genetic mechanism in those microorganisms that contributed to the resistance to these herbicides? And my third question, if I may, it's related to the use of herbicides. We know that they really have a negative impact on the environment and on the insects. But you know, in Africa, especially in sub-Saharan Africa, farmers are facing issues with weeds, and including parasitic weeds, which I'm working on. And farmers really have to rely on these herbicides to control these parasitic weed species, because we really don't have any other ways. So what do you suggest in this case? Because the herbicides are affecting bees population, but at the same time, farmers are facing issues with weeds. So what do you suggest? Thank you.
So the 20% survivors, yes, it could be also that the genetics of the bees plays a role, but then we would not expect, thank you, to see differences also between the different pesticides. Yeah, so, but that is something that could be addressed also by testing different colonies, different genetic origins of bees. Good. What was the second one? Oh, the resistance to the herbicides. Okay, that is a bit difficult to say, because we had initially chosen these generations that we sequenced like more or less equally distributed over these cohorts. But the most important things when we see that there is some kind of adaptation to the herbicide happened very early, and we have unfortunately not sequenced those. So this is what we would need to do to understand better what was the changes in that small window. Yeah, because we see now the bigger picture what happened over the whole long time period. Although, for example, in paraquat we already see that that initially there was a huge reduction, and then two more taxa came in. Whether this is then the reason for that they improved in survival, that we could be, but we don't know. So we would need to have these other groups being sequenced. And when the herbicides are actually needed to be used, because there is a very critical weed that you need to get rid of. Yes, then from this we know the herbicides are can be detrimental to the bees, and we know that people need to use them. So then it's a question of, for example, how to apply and when to apply. So bees are not around 24 hours of a day. So they have certain times during the day when they are foraging. Yeah? And they start fairly early in the morning, but they also they stop foraging at maybe late afternoon at around 5 o'clock. And the best way, that is also what always was being advised for insecticides, is that farmers should spray rather in the evening, because then there's a long time period until next morning that pesticides have time to penetrate the plants, so that the liquid that they're applied with evaporates and so on, so that the bees are less exposed when they are foraging at the next morning to those pesticides. So that means their dosage that they pick up is much lower. And that is something, actually quite easy mechanisms that could help already to protect bees and also other beneficial insects. I think there was another question.
Yeah. Thank you.
Thank you very much. Thank you very much for that interesting topic on the importance of bees. And I also want to borrow from one of the statements we have just mentioned, that we don't want to mention the manufacturers of those herbicides. And very curious about why Africa becomes a dumping site. I don't know why you want to kill all the bees in Africa. What next? You kill the bees, Africa becomes hungry. And Africa remains dependent on foreign food and importation. I want to commend you for recognizing the importance of the microbiome. Bacteria are everywhere, including human beings. Today, the recent studies are on the metabolites of the microbiome on the human gut in relation to lifestyle diseases. One of them, cancer, esophageal cancer, what colon cancer, and the like. So, bacteria are useful and very important, my friend. Finally, I don't know whether this rush towards biopesticides have been tested on their influence on the life or the life of the survival of the bees, because nowadays we are rushing towards use of biopesticides towards controlling the pesticides. I don't know what's the impact and what has been the implications on the population of bees. Thank you very much.
Yeah, so the thing with Africa being the dumping site for pesticides is indeed a problem. Yeah? So you see what is happening. Companies have produced amounts of pesticides and then they're banned in Europe, and the companies think, hey, we have them here, we can still make money. And then it comes another point, is that often in many African countries, the regulations are not very strong, they are rather weak. which pesticides are allowed, how they are registered. Is there a review needed so that registrations can be extended? And so there's also a lot of weak policies around pesticides in many countries. And that is what these big global players, what they simply exploit to make profit. That's, I think it's a lot that is just about money. Good, so, okay, so, what was the other thing? Biopesticides, yeah, that's also, so they are pesticides. So they still kill things. And for many, it has also been shown that they have some effects on beneficial insects. So beneficial insects is not only bees, yeah? So there are also many others, predators, parasitoids, and so on. So there's more than bees that are beneficial. And some of them, they have also an influence on them, and others not. But the testing of this is it's rather weak. So often it's just the claim, oh, it's biopesticide, it must be good, and then we don't need to test it. So we have done this in the past with a few entomopathogenic fungi that are used, and we could, for example, show that at least those 5 that we use, the bees are safe from them. But there are plenty of different products that have a different basis, and many have not really been rigorously tested. There's often just the claim they are good. There's a need for more investment into that area. Thank you.
Thank you. Any other question? Okay. Thank you. Once more. Another one.
Okay. This is the last one. So I'm thinking, what about us as the consumers of these? Is the effect the same? Because sometimes I know we do wash maybe some fruits when we spray them with herbicides. But could it be happening to us as well?
Okay.
Potentially, yes. But it is more easy to do experiments with bees than to do experiments with humans. But we could do this. We can just split it here into 2 halves, and we have a control and an experimental group, or the other way around. But I'm not so sure whether many would agree with that. So it's more difficult to study here. There are also more different complex factors that we need to consider, yeah, that play a role. But I'm pretty sure that lots of herbicides are also not good for us. And there's one that is always has made it long time ago already in the media, that's called atrazine, that is a herbicide, but that has been shown in, at least in mice or rats, to have really negative effects, where also people assume that it might have similar effects on humans. So I'm sure maybe very good washing of your vegetables is needed.
Thank you. Let's applause Professor Mike. Yeah, so the next presentation, and perhaps the last one of the day, of today, is from Joan. Yeah, Joan Kiplagat. Yeah, from the University of Eldoret. Yeah, she's going to present on genetic breeding in silkworm, Bombyx mori, for environmental adaptation. Yeah, Joan, welcome.
Good afternoon to everyone. Yeah, my name is Joan. I am a student at University of Eldoret. A PhD student. I'm doing research on Bombyx mori, which is a silkworm. So in this work, I'm presenting a review. Yes, we did a review on genetic breeding strategies for environmental adaptation in Bombyx mori silkworm. And so that's the Bombyx mori silkworm. It is an insect of complete metamorphosis with the egg, larva, pupa, and adult. So the pupa is the cocoon. And this is just like bee, it is an insect of economic importance because it produces silk. The silk from the Bombyx mori is rated as one of the prestigious silk, and that is why this worm is being reared. So the sericulture was not developed in Africa, but rather it was introduced to Africa. The origin of sericulture is China, so it was just introduced to Africa sometime, and in Kenya it was around 1972. So the part or the stage of the Bombyx mori that produces silk is the pupa, which is encased in the cocoon. And it is that cocoon that produces the silk. And this worm has undergone a lot of domestication. Its origin, or it was rather developed from the wild moth called the Bombyx mandarina, which is the wild moth. But because of the interest in the quality of silk, it has undergone domestication to the present day form, which is completely domesticated. And in the process of domestication from the wild form, there was a lot of gene manipulation and editing so as to make it more resilient to environmental fluctuations like humidity, temperature, and the stuff. So So I've mentioned so much about the moth itself, or the silkworm. Now, so it is this fragility, the domestication that has made it so much fragile to adapt to, if it is exposed to fluctuating environmental conditions like humidity and temperature, then it won't survive. Why was the gene manipulated? As I said, it was manipulated to increase the quality of the silk. And the methods that were used before were the traditional methods that were focusing on fecundity, thermotolerance, larva survival, and cocoon weight. And that resulted to the production of hybrids. Those methods, though, were good and developed the breeds. It was time-taking because it is long, crossing one and another to produce and picking the superior larvae from the outcome. It was taking a lot of time. So the genetic breeding for environmental adaptation is therefore more appropriate in picking the genes directly from the worms rather than crossing and picking the superior ones. So in this selection, in this molecular work, silkworms, molecular tools like marker-assisted selection has been used to identify the quantitative trait loci for stress tolerance. And as I said, the importance of that is that it takes a shorter time and enhances the genetic gain. There's also the use of transcriptomic in the expression of stress-responsive genes, which has also been used. And pangenomic comparisons has also been used to identify structural genetic variation and to improve the robustness in the silkworms. Now, the objective of this review was to consolidate current information on genetic breeding strategies for environmental adaptation, and as well as to synthesize and find the previous used conventional selection methods in the development of the breeds. The method that was employed in this review was systematic and structured approach, which consolidated the evidence on both classical and modern breeding. And the literature was conducted or extracted from legitimate sites, which include the Web Science, the PubMed, and the keywords that was used in searching for these. The keywords were select breeding strategies, gene editing. Those were the keywords that were used, and the search time was between 2015 to 2025. So any articles that fell in between that duration with the keywords were picked. And as I said, to ensure reliability of the evidence, each study was assessed using quality appraisal criteria adapted from PRISMA guidelines. And I'm going to give some of the results because of time. I have so many, the list is so long, but I'm going to highlight few results from the search between the 2015 to 2025 on the breeding methods that were applied in these. The first one is a research by Singh et al. In 2019, which was done in tropical India. And they used the line tester analysis targeting the trait on heat tolerance. And the outcome is they identified the hybrid which, with high cocoon yield under the temperatures of 36 degrees Celsius, which is way above the optimum for rearing, which is around, the optimum is normally around 25, and if it goes, should not be beyond 28. So that was one. Another study by DOS, still on targeting thermotolerance. They used dialysis cross, and the study was in India, resulted in crosses that were also able to survive the stress of beyond 38 degrees Celsius. And on fecundity, I picked Wang et al., the study of 2021, which it was done in China, and they used pure line selection, and they were targeting the bovine strain to increase the number of eggs. And the result was an egg which was able to lay over 500 eggs. Normally, the female moth lays 350, but the outcome was increased fecundity with around 500, which is good. And on hybridization and survival, it was a study by Ali, which was done in Pakistan. And of course, survival is still key in sericulture because the more they survive, the more they yield. So it resulted to hybrids. Their study, the outcome of the study was hybrids that were developed, which were able to survive. They had a higher rates of survival by 20% on fluctuating relative humidity. So when we talk of temperature, I said, what does it affect? It affects the physiology of the worm, same like the relative humidity. Maybe I mentioned something on relative humidity because this is an invertebrate. The fluctuation in humidity, when the humidity is extremely low, it causes evapotranspiration in the worm, causing dehydration, and the general physiology of the worm is affected and the celiac outcome. So the relative humidity should be stable to reduce that evapotranspiration from the worm. On cocoon weights, Kumari et al. 2020 also did a study. And the breeding method they employed was backcrossing, which was done in monsoon zone, and it resulted with hybrids with higher cocoon weight of dias being 1.5 gram on average. Most of the cocoon weights are around 0.98 there to 1 gram, some to 1.2, but 1.6 is higher. And the importance of the cocoon weight in sericulture is the higher the cocoon weight in most cases, it means the higher the silk that is that will be extracted from the cocoon. Then filament length also is key, and a study by Sharma et al. 2023. They looked into the breeding method is recurrent. They used recurrent selection, and the outcome was long filament. Long filament is good because the longer the filament, then the quality of silk is high. Because when the filament is long, then it can span a continuous fabric, unlike the one that is broken. So it produces an even fabric. Another study in semi-arid India on by Yadav on feed conversion. They are targeting feed conversion because the more the mulberry or the proteins in mulberry are converted to feed, the more the silk. And the breeding method is, the use is feeding tests and progeny, and they identify the strain with the highest survival under drought stress. So we have, there's so many studies there, but because of time, maybe I'll just mention which one. Now, maybe I can mention 2 from here because there's so many. And in Japan, there was a study on transcriptome analysis, which was targeting humidity stress and also increased pupation also. It was done in the year 2020, but Wanabe and et al., it was a group of scientists who are looking at, the outcome was the downregulation of critical protein genes under high relative humidity. So that resulted to increased pupation. So those are some of the studies that we looked into and concluded that genetic breeding is a critical, sustainable strategy to enhance silkworm resilience against increasing climate volatility, including heat stress, humidity fluctuations, water scarcity, and declining mulberry leaf quality. Mulberry is the sole food for the Bombyx morus. Quality of mulberry is equally important. When it declines, then it also results in, it affects the quality of silk. And then also the classical approaches, that's pure line selection, hybridization, remain the backbone to successfully producing regional adapted hybrids with enhanced survival, cocooning, and thermotolerance. And another conclusion on this study is that marker-assisted selection and QTL mapping accelerates early precise identification of loci linked to stress tolerance. So the use of the marker-assisted selection and QTL is key and in identifying those specific genes. And lastly, genome selection enhances precision for polygenic traits for leveraging all genome data. So the gene editing platforms like CRISPR-Cas9 also offer precise manipulation of stress-tolerant genes, unlocking opportunities for rapidly growing climate resilience and quality silk. And the recommendation for the, from this review is incorporation of GS or genome selection into conventional pipelines to increase the accuracy and efficiency of selecting for complex traits early in silkworm life cycle. Application of precision tools like CRISPR-Cas9 to directly target key traits, 3 key stress genes for rapid improvement in thermotolerance and silk quality. So SP70, so those are the genes that are linked to thermotolerance in Bombyx morus. And another recommendation is to tailor breeding efforts using agroecological mapping and GG plots that that perform optimally and stable under local environments. So this brings together experts in genetics, agroecology, and climate resilient policymakers to translate these advancements into practical gains for sericulture, application and adaptation for economic improvement and increase in silk yield. Thank you.
Thank you, Joan, for that good presentation. Are there any comments or questions for Joan?
Thanks for your presentation. The studies that you showed us in that table, and you said this is just a snapshot because there's much, much more. But at least what you showed us looked like it's all happening in Asia. So can you tell us out of the total, is there also something like this going on in Africa?
In Africa, there is little work done on this. I also to do the same because I'm researching on very silkworm. I was also to compare among the strains because I'm working with silkworms in different agroecological zones, high, mid, and low. But the constraints of the markers, the primers, the technology we do not have. I really wanted to do also, but it was very difficult. And if I see the studies that have already been done around here, there's very little. There's very little, yeah. So that's why I did a review on this, because I was told, I estimated the cost of some of the reagents personally, because we had to do with my supervisors, and it was coming to almost like 300,000, which is high. So maybe that's the reason why there's little on that. So they just do some generalized form of studies, yeah. And maybe the Asia, it is biased towards Asia because that is, it is native. I think that's where it originated, and yeah, it is more attached to that. Still, even in Kenya, there is apart from Isipe and Karo, very few farmers are practicing the sericulture thing, despite it being possible.
I think Karo with the sericulture unit, they
Yeah.
do this together with these Japanese people.
Yeah.
Because there is this idea to bring silk production to Africa.
Yeah.
Because like in Japan, there's no silk production any longer.
Yeah.
Because it's too expensive to do it there, and they cannot compete against the cheap Chinese stuff. And I think the India, Japan, they are interested in exploring Africa and the opportunities. And I think that is because the conditions are different in Africa. So that is a good thing to maybe also step into this, looking at the silkworm adaptation, but also the mulberry side. Yeah, the mulberry has to grow here. So I think that opens up quite some opportunities.
Yeah, under the climate smart agriculture, that's what it is coming up right now. Apart from, as you said, apart from Karo and Isipe, so and the agenda of being introduced to Kenya by the Japanese, it was a collaboration between the Japanese government and Kenyan government in 1972. Actually, it was to increase the yield. The conditions for growing mulberry in Kenya is good because there's no winter, you know. During winters, that's where there's a break of production, but there's no winter. So if the conditions, the room conditions are regulated and the mulberry can grow, actually in my study when I was doing a survey, mulberry is growing almost everywhere in Kenya. So sericulture is an opener. There are so many opportunities and so many companies from last year through this year are coming to explore, to see the possibilities of that together in Kenya and also in Uganda. They are coming to Kenya, to Uganda, and to Tanzania. So I agree it is an opportunity, and I've also been invited to so many forums to give the insights on the same and to discuss the possibilities. So actually there's a lot of possibility if much of research and the study of these is extended mostly on how if we look at how they are adapted, so which are the genes that are targeted, then we can specialize and produce genes that are the strains that are more adapted to that specific region by targeting the genes. Then we can have being reared across all these sites of Kenya, and it can be of global importance. Yeah.
Okay. Nothing else? Any other comment for Joan? Maybe I just make a general comment. There was a strong push to produce silkworm, especially in Western Kenya, around 2000, around the year 2000 there. There was a strong push and many farmers had gone into it. But I see that most of those farmers have either closed or switched to something else. But I think that even the market and the market for silk perhaps could be a challenge, and it could be discouraging the farmers who want to really produce silk.
Yeah, the one in
That's
Kakamega,
just my imagination.
Yeah, the one that was introduced to Kakamega County, it was targeting women and youth. Yeah, it collapsed because there are so many constraints that come there. For it to be economically viable, volumes is required. So a lot of mobilization needs to go into that. And once there is increased production, so when so many people produce, then it becomes easy for it to be shipped to Japan. So otherwise, if so few people are producing, then putting up the machines and the staff to do it becomes a problem. So the mobilization part and the surety that the breeds will survive, because also it is highly sensitive. The rearing practices needs to be the ones that will increase the size. Otherwise, it can really die, and you have very little of them. So there are so many things, but I believe, as one of the researchers in sericulture field, if we call the mobilization, the more research, the more sensitization, we'll get there one day. And also the river tax here. You see, also river tax is, if our textile industries could hold into that, then remember this is it's a natural fiber. So when it comes to climate change and sustainability, it is really one of the fibers that we should be targeting as far as environmental pollution. And on the mulberry part, it is also good for environment. So it is a score if we are.
Okay. Okay, thank you. Let's applause Joan. Thank you. So that brings us to the close of today's session. I thank all of you for your patience and participation. So I'm told that tea is ready, so we will just get out and take our tea. Then another general announcement is that there will be dinner, I think, from 7, and dinner is for everyone, so don't leave before dinner. So after tea, you can...